analytics with safeguards, we suggest to risk the uncertainty, but we allow humans to leave safeguards every time they risk the uncertainty.
Linking and brushing is very useful for interactive video data exploration. Data-driven brushing tools are getting popular recently and achieve good results, for example the Mahanoda-based brush and the CNN-based brush. However, these brushing tools are optimized by the data from a large number of users, which are not suitable for everyone. In this paper, we introduce an adaptive brush model which takes the user in a loop to improve the brushing accuracy.
A testing environment for continuous color maps. Many other fields in the computer science do it, we should do it too. With our work, we introduce the approach of using test functions as a standard evaluation method. We present a test suite for continuous color maps implemented in the CCC tool. Adapt the test to your requirements at the interactive testing section and observe them at the testing evaluation section.
All right, Joy, am I good to go? Okay. Good morning, everybody, and welcome to the, oh, this is the wrong presentation. Yeah, okay, wow, that's a good start to this morning. Good morning, everybody, and welcome to the BioVis Challenges presentation. I am going to see if I can get the proper presentation up because this is 2020 and that's exactly how this morning is going to start. I'm really happy to have you here for our first virtual BioVis ever. It has been, of course, an unusual challenge in planning for today. Okay, it looks like it completely deleted itself. That's great. Nope, there it is. Okay. Good morning, everybody, and welcome to the BioVis 2020 challenges. My name is Anna, and I am really happy to have you this morning, finally, with the correct presentation. Hopefully that's the only snafu today. We would have loved to welcome you in lovely Utah, but thanks to 2020, we are welcoming you for our first ever virtual BioVis. So for those of you that are new to it, BioVis showcases new and ongoing research in computational biology and computer science. And we try to highlight some of the problems that arise in biological data visualization. And we do this by hosting this cross-disciplinary forum between experts that work in biology and in healthcare, as well as with data visualization experts as well. So BioVis has two events that are co-located with two major conferences. The first is BioVis at ISMB, and this is a community of special interest. It is our larger event drawing often between 200 and 300 participants each year at the ISMB conference. Our challenges workshop is this one right here, BioVis at Viz, where we integrate people from the visualization and the biological community to work on different problems and to present different problems that they have when visualizing data. So BioVis this year was virtual in both senses. The BioVis happened in Montreal in July of 2015, and this one, of course, is happening in Utah. I just want to give you a sense of the day of what we can expect. We're going to start off with domain expert presentations. These are individuals who work not in data visualization or do data visualization research per se, but who often use data visualization in their work and even build visualization tools themselves. And they're going to be talking to us today about their projects and their processes and some of the challenges that they have faced. At 10 o'clock, we're also going to hear from our data and redesign challenge folks. These are submissions to BioVis answering the various challenges we posed through the data challenge, as well as redesigning a visualization that they felt needed to have another look. We're going to wrap up with the capstone presentation as well from Niels Gellenborg. I want to take a brief moment to introduce all of our speakers, and I really want to thank everybody who submitted or who accepted the opportunity to give a talk today. It means a lot to us to be able to put this program together. So we're going to start off with David Aanensen, who's joining us from the Oxford Big Data Institute. It will be followed by a talk by Gus Dudas, who is a freelance consultant and a colleague of mine when I was hanging out at the Hutch Institute. And finally, we'll wrap up with Sidney Bell, who is a computational biologist at the Chan Zuckerberg Institute. They've all got really excellent presentations about their experiences using BioVis in their day-to-day, and I'm really, really looking forward to their excellent presentations. Hilary Coon is also going to give a talk introducing our data challenge. Hilary is joining us from the University of Utah, and she will be followed by two data challenge submissions and 2 redesign submissions as well. And finally, we're going to wrap up with Niels. One special note about Niels is that he joined us from the founding of BioVis 10 years ago. And so he's going to give us a little retrospective on how things have changed over time. And so with that, I'd like to turn the mic over to David so he can talk to us about his experience with biological data visualization in these COVID times.
Thank you, Anna. Can you hear me okay?
Yes, I can.
Great. I'm going to share my screen, so I'll ask to see whether you can
Go ahead, David.
Thank you very much. Firstly, thanks, Anna, for the invitation to this conference. I've watched the conference very carefully over the years, and it's a real pleasure to be able to present at reasonably short notice today, but thank you ever so much. So my name is David Aanensen. I'm going to talk about some of the work we do at the Centre for Genomic Pathogen Surveillance, which is currently co-housed between the Big Data Institute in Oxford and the Wellcome Genome Campus, home of the Sanger Institute. So we work, me and my team, in infectious disease epidemiology, and one of the key challenges when we're looking at any sort of disease paradigm is how to capture information about pathogens, where they are, what they are, when they were sampled, what kind of disease they're causing and who they're infecting, and actually then untangle which cases are related to each other so that we can try and look at directionality of who's infecting who. And I think we're all acutely aware of the importance of this kind of approach over the past 6 to 9 months. So we use whole genome sequencing, and this is a method that's taken a lot of uptake everywhere over the past number of years, which gives us the unique genetic code of every isolate of a pathogen that's genome sequenced. Now, one of the things we want to be able to do broadly, collectively as a community, is to be able to understand this kind of transmission at very local levels. So can you understand who's infecting who in a hospital, or foodborne outbreaks of disease through food production plants or in water sources, etc. And then can you scale this up to looking at between these institutes across a country, and then ultimately upwards towards spread within a continent and ultimately globally. And I appreciate the irony of linking the UK to Europe here. I'm firmly of that mind myself, but you know, I can't talk for the majority of the country at the moment. But ultimately, this is we need systems and the ability to be able to monitor data at this kind of scale. Huge amounts of challenges in very different areas. One of the approaches that we take, you collect a bunch of pathogens, you run them through a genome sequencer, and then one of the ways that people represent the similarity between lots of samples within a study is by representing them as these kind of trees, this kind of cartoon nomenclature that shows how similar things are to each other. And this gives us a really nice unit of surveillance, which is the difference between any two genomes or strains within the system that we're focusing at, which can help us understand this kind of transmission process. And also, if we're looking for the presence of specific genes, or if you have one sample that has resistance to an antibiotic or a vaccine versus another, looking at presence or absence of specific genetic markers can help. Now, there's a huge amount of information and data on both sides of this equation. Think of all the information that one would want to capture about time, place, space, host, etc. on the left-hand side, combining that with the genomic data. So one of the things that we try and approach is how do you distill that information into usable visual formats that enable somebody to be able to overlay their expertise and pull out hypotheses from the data, this kind of individual in the bottom right here. And this individual could be sat at any of these data scales. So it could be somebody who's in charge of infection control in a hospital, or somebody who's in charge of kind of quality control in a food production plant, all the way upwards to national public health institutes, or European, or the US CDC, for example, and then ultimately upwards towards the level of global analytics. So just some kind of very basic. So this is the kind of approach that happens. So samples of these bugs are taken, they're run through their sequencers, and then you get the genetic code out of the other end. And this is a standard way of comparing those genetic codes. So you can see that this is representing 3 different samples or isolates of any pathogen. It could be a virus or a bacteria. And then you count the number of differences between each of them. And then this can be very easily visually represented in these kind of dendrograms or trees. So in this case, 2 and 3 are more similar to each other than they are from 1. So they are co-located on this branch here. And then we might want to overlay pieces of information that we know about the samples that have been taken. So in this case, do they have, can they be treated with certain drugs, for example, or any kind of information that you could visually represent as blocks next to these kind of tree representations. And then if we have information about geography, for example, we can plot that on a map and compare how the similarity of the genomes look compared to where they're sampled from. So you can see here, 2 and 3 are both genetically similar. They have more of the things that we've mapped against them in common, and they're also been sampled from the similar location. So this is building up our hypothesis to interpret this data by using the visual interpretation. And then if we have temporal information, so were they sampled within a very small time span? And if so, can that help us bring out our hypothesis and understand what's going on within this scenario? So again, this very simple timeline here, 2 and 3 are sampled within the same, very close to each other. Their genetics are more similar. They have more in common from their additional data, and they're from a similar location. So this points us towards the, building hypothesis about whether this was potentially a transmission, and we overlay all the other information on. And this is how many people who work in the field of genomic epidemiology use these kind of information and to try and understand systems under investigation. This is a real world example from a set of MRSA, MRSA genomes from across Europe, where there were 3 major geographic clusters that tend to cause more disease. And you can see them on the map in Portugal, Spain, and Germany, for example. Very, very similar kind of data processes that happen regardless of which organism or pathogen you're looking at. And of course, these trees can be represented in different ways. So these are different layout algorithms for the trees. They don't change the relationships between the leaves, but they can help our visual hypothesis come to life. So for example, this bottom representation here is kind of quite a similar shape that we see fairly frequently, which is kind of what an outbreak looks like, where you have kind of long branches at the background, and then you have suddenly some very short radiation that indicates that something spread very rapidly. So within our center, we build tools that try and sort of address those processes of collecting, analyzing, processing, and delivering data, so that individuals can try and understand and bring their own expertise, depending on kind of where they sit, towards this information. And we work both together with many researchers, but also with major public health agencies. We also work with laboratories to build capacity in genomics and actually, learn in all directions about actually what are the kind of questions that need to be pulled out of this kind of information, and can we help build that back into tools and visualizations that help researchers and public health individuals make decisions. So 4 general tools, I'm going to show you 1, possibly 2 of them, and they're all focused around data collection, analysis, linkage, and visual analytics. Some of them based on processing raw genome sequencing, and the others on collecting metadata and linking it together. We have throughout our applications the philosophy that we try and identify things that are horizontal data and visualization processes that could be applied to many, many different kind of vertical processes or species, etc. So I'm going to do a demo of one of our tools. This is a tool called MicReact, which is focused on bringing visualizations to people's data that they bring to the system. So if you have data sets that consist of these kind of trees and clustergrams, and you have information about geography in there and time, and maybe you have network diagrams that you want to bring together, how do you just bring that together very easily and intuitively for explorative purposes? So this is a tool that's been used in many different locations, and we have many example data sets about there, and I'll show you a couple before. I know that Gytis is giving a talk afterwards talking about how terrible trees are. So this, I hope this doesn't set up nicely. But for example, this is one of a piece of work that Gytis was involved in generating. So these are the West African Ebola epidemic information from a paper. So this is you can see the tree, which represents the genomes of the samples and how similar they are. And then moving along, you can see the distribution over time of where these genomes are. It's very simple sort of interface. We have a map, we have a tree, we have a timeline, and you can interact with each one of them and sub-select in each one, and it highlights pieces of information on the rest of the data. So, very simple interface. Many, many examples there, not from viral, bacterial, and other paradigms. Now, I'm going to show how we can create one of these very easily with data that we've produced ourselves. So I'm in my group, in my lab, I've sequenced a bunch of something, and I've got a nice tree, and I've got lots of other metadata information, and I want to build one of these visualizations around them. So in this folder, I've got 3 files. The first one of which is a metadata file that contains, for each of the samples in my data set, some set of information about them, a name, a latitude and longitude of where they may have come from, And then some other pieces of information that I'd like to understand how those pieces of information relate to how similar the genetic material is. So I can fill this with many different columns of information that I may then want to overlay with different colors, etc., etc. And then I've got two other files in this folder. The other one is what's known as a Newick file or a tree file. Now this, nobody ever really needs to see this, but this is an industry standard textual based way of representing branching patterns on a tree. It's a set of concatenated brackets that is produced by sort of any any program that produces a tree format. And then the last file I have is a network file. And again, this is just a standard industry format, dot format file representing nodes and interconnections between nodes. So I can load one or any of these files into MyCreate by dragging and dropping them and then giving my project a name. And those files are then uploaded and linked. So the information in the metadata file, the latitude and longitudes are pulled out and place the points on the map. All of the other information is placed in this table here. The day, month, and year is used to be able to build a very simple timeline. And then the Newick file, the tree file, is represented here as a phylogenetic tree. And then because we had a dot file, we also have the ability to turn the map off and look at the network file. And this has some information that will help me understand my data set in a much more intuitive way. So let's go back to the map. I can interact with this in many ways. As I mentioned, I can sub-select a single part of the tree. I can use a lasso tool to very simply move around, which will highlight sections in the other visual windows. I can do the same with the tree. I can sub-select single or multiple nodes that I can use lasso tools, etc. I can overlay all of the other pieces of metadata as these kind of blocks that may give me some better understanding of what's going on in each of these subsections. And I can, of course, change the shape of the tree to different layouts to help me interpret what's going on. Similarly, on the timeline, we can play back and forth, as I showed with that example previously. But maybe I have a better understanding of this data, or one of my colleagues, and I can share this information with them and they can try and understand. But I spend a little bit of time pulling out that actually this is the piece of information that's most important. And it's most important to highlight this particular branch of the tree for whichever reason. So I can then create also a link that allows me to save that particular point in the process and send this link to a colleague and say, look, this is the piece of information that I think we need to take some course of action on. And maybe I also want to add a note that allows me to say, okay, this is what we need to do. And I can save that, resave that link, send this to somebody upwards in the information chain who needs to, or has the capability to make some decision. And then they can simply open it up in their browser when they receive it. It jumps them straight back into that space where I've inflicted my expert knowledge on top of the data that's been presented. Okay. Okay, so this system was up and running for quite a long time, and we've been sort of refining various visualizations, etc. But of course, the pandemic hit at the beginning of this year, and we had to really kind of start to scale up, because trees of sort of reasonably small sizes are fairly easy to handle and are very intuitive things to be able to represent this information. But when you get very, very, very large trees, firstly, the value of a very, very, very large tree becomes slightly questionable, but it does provide an incredibly good framework for then delving down into these subsections. So I was lucky enough to be involved with a big project in the UK, which the aim was to sequence every positive COVID case, so SARS-CoV-2 genomes, the name of the virus. And to this date, there's been roughly 140,000 SARS-CoV-2 genomes sequenced, and that makes a bloody big tree. So we've had to really re-engineer to scale up and really think about how we can deliver this intuitive information a bit better. There are lots of challenges still there. So we work with public health agencies in the UK and elsewhere to try and deliver these kind of tools and information to allow them to link their own data together. So this is just the sheer increase in the number of genomes that have been sequenced for SARS-CoV-2. And I would go so far as to say it's probably one of the most sequenced species on the planet now. I mean, it's important for us to understand whether this information can be used to help interventions and understanding the spread of this particular pathogen. So the consortium I've been involved in consists of many institutes across the UK. It's a truly decentralized collaboration where sequencing sites around the UK are generating these data on a daily basis and feeding in to the global databases. So we've had to just simply to be able to display trees of these size, we've had to go through a re-engineering exercise at two specific points. One where we reached about 50,000 genomes, and that's a pretty big tree in the first place. But now we've passed a sort of a somber milestone of about 120,000 leaf tree. So we had to rethink again. It gets much more complex each time. But also, what's the value of looking at a bloody massive tree of this size? I mean, aside from giving a really good framework to be able to delve down in. Now, these big trees need to be described in much more useful units or nomenclatures that I could speak to somebody on the other side of the world about a specific component of that tree. And there have been a couple of lineage schemes. So this is essentially breaking down the big branches into smaller, more manageable branches that can then be discussed very easily. So, for example, somebody in my location has turned up with lineage A.1. So I can describe that and somebody else knows exactly what that means. It means a subset of the tree. It doesn't necessarily have any different biological behavior, but it just breaks down these very large trees into manageable units that can be talked about. So this, for example, is the current tree. So on a weekly basis, we push out another version of this, and it's automatically generated from the COG UK consortium. There's a central server called the CLIMB server, where all the genome data is deposited, analysed by phylogenetic pipelines produced by groups like Andrew Rambow, etc. And then we push this visualization out each day. So this tree is 116,000 leaves. And we've had to move from, within a browser, how do we produce an intuitive experience here? So going from using sort of Canvas to actually looking at optimization of how we can actually bring this information together. But also, we've got the tree in the first place, which does allow users to move in. But why can we not produce different visualizations that, for example, this here describes those sets of lineages. So it breaks down the tree into much more manageable units and then shows when those units first arrived and when they came. And of course, you can then update each of these visualizations by sub-selecting parts of the map, for example, or searching the metadata to, for example, only look for lineages that have occurred in a particular part of the world. And this is some of the ways that people are using this information to try and understand scenarios. And then who's to say that there aren't better ways of visualizing this information? So this is a bespoke visualization that was built. So we've incorporated a library that allows people to write their own code for producing visualization. So we use the Vega open source library, which is very useful, which allows people to amend and adapt visualizations that will sit on top of the data in a bespoke fashion, and then they can save those and share them in the same way that I described earlier. And then we can also overlay additional pieces of information. So this is a global tree because all of the global information sits behind it. It's not just UK focused. It shows the distribution of a particular mutation in SARS-CoV-2 that has raised some concerns, but doesn't seem to be indicating any specific biological material. But you can see quite nicely that from the beginning of the pandemic, most of the strains had a particular variant of this mutation and then moved to the second through. It just gives us this intuitive way. So one of the things we've tried to push is not just this kind of open public visualization and linkage of this data, but also deployable local versions. So MicroReact has been deployed in public health agencies. And then one of our other tools, Dataflow, which really enables very visual ways of bringing data in from different sources, transforming it, and then having a visual pipeline of how that data is analyzed before being pushed into visualization tools like MicroReact. Now, this allows very resolute information such as postcodes, PII, patient identifiable information, to be used within agencies to be able to, again, bring their kind of intuitive visual hypothesis together to try and use the data to make interventions. And this has been used in agencies such as Public Health Scotland and Wales to really understand where these kind of lineages are spreading, and to use that information to filter up to governmental level. So in Scotland, the first health minister has been given this information, and here's a picture of Jacinda Ardern in New Zealand, the Prime Minister, being shown the lineage distribution across New Zealand using microreact. And what's nice about New Zealand is they have been able to genome sequence every single positive case of COVID-19, SARS-CoV-2, the genome. So they have a really valuable, intuitive way of bringing genomics to bear into such a crisis. And they have used the trees to be able to make decisions on lockdowns. I was going to show you a couple of other things, but I think I've gone on a little bit longer, so I'll just stop there. But I just want to say that thanks to my team for producing such wonderful tools, and we'd love to hear thoughts and advice about how to do these things better. Thank you.
Distance of 0 to each other.
So I should say that we didn't. I should say this is a global effort. In the UK, I think roughly about 70,000 of those genomes were sequenced from groups across the UK, but it's a true global effort. Groups across the world are attempting to sequence as many of these genomes as possible, because that gives us collectively, globally, a much better understanding of what's going on. And it's a truly collaborative global effort. Now, off the top of my head, I couldn't give you the exact number of genomes that are identical, but it's very small. SARS-CoV-2, the genome, it evolves very slowly, so there aren't huge amounts of variation cropping up. I can't, off the top of my head, remember the mutation rate, but it's much lower than other viruses. Gytis maybe has the figure off the top of his head, but very few of them are absolutely identical.
That's fascinating. I also have a question about, as you talked through the challenges of getting so many genomes, especially in a pandemic where there is a lot of worldwide effort to get all this data into one place. What has been the biggest challenge for you? Has it been, getting the data? Has it been processing the data? Or has it been that visual end where you're trying to communicate it out?
Well, I can give you my particular perspective. I mean, this, again, there's a huge amount of very, very good groups working on this. There's an international open data source called GISAID, which acts as a repository for anybody to be able to submit genome data that's kind of been produced. Now, within the UK, the COG UK exercise, because the genomes were produced from labs and there's a central kind of data analytics server, this was kind of fired up in about 2 months, a really kind of strong collaborative effort to really build the data pipelines to not only bring the genome data into one location, but also process that amount. Really producing these trees demanded a huge increase in the algorithm optimization, and Andrew Rambow and his team in Edinburgh have done quite fantastic phylogenetic work to be able to ramp up the pipelines. From my personal point of view, we were trying to push out the visual analytics on top. It was really just coping with the scale, really thinking about, okay, so in the past we've always talked about how we're going to be able to visualize trees of a couple of 100,000 nodes, but we didn't really expect to reach that in 4 months after a particular incident. So we've had to really the engineering team have really had to think about now, how do we bring a 1 million leaf tree, and what's the use of a 1 million leaf tree, and actually how do we bring that in a way that maintains that kind of visualization. So there's been some optimization methods, but also some new technical challenges, looking at new technologies like WebGL and how we implement that rapidly so that we don't have to worry about re-engineering in another 6 months when the doubling time of data increases. It's been both a very somber and sobering period, but also a technically challenging piece.
That's fantastic. The 1 million node tree or the 1 million leaf tree, that's absolutely fascinating. But yeah, we are there.
Yeah, I think honestly, I don't think, I think some of the visualization challenges have been solved with respect to how we bring those kind of scale of trees together. I mean, the key is how do you pull out the pertinent information for that person who's trying to understand that tree, and that person could be, at a hospital level, or it could be somebody who's in charge of a national control program, or it could be up to the WHO. So there's different stakeholders that need different distillation or pull out of the information in those trees to be able to make visual interventions almost, I think, yeah.
All right. Thank you so much, David. I can give you like a round of applause virtually.
That's like a silent one-hand clap. That
yes.
Brilliant. Thank you for your time.
Thank you. And so keeping on the theme of trees, we will now move on to a presentation by Gus Dudas. This one is recorded, Joy, and he's going to tell us how trees are phylogenetic trees are a technically true visualization disaster.
Hi, my name is Gus Dudas, and I study how viruses evolve, and through their evolution I try understanding how viruses spread and how we can best design interventions that can stop them efficiently. So during my work I get to wear a lot of different hats, all of which I'm relatively uncomfortable with, but if someone were to call me a phylogeneticist, I probably would mind that the least. And even though I love producing phylogenetic trees and I love visualizing them, I also think that they're a fundamentally difficult data visualization technique, which I will try to explain during this talk, and also show you a couple of examples of how I tried solving that. So right off the bat, I have to warn you that phylogenetics as a discipline is pretty esoteric. And that is primarily because the methods that we use to produce the phylogenetic trees don't really know what sort of data you give it, if it's bad quality, or if the models that you're using are completely inappropriate. It will still give you a phylogenetic tree out. And so doing good phylogenetics is actually only possible by a relatively small group of people in the world. And similarly, because of what I'm about to present, phylogenetic trees can also be misinterpreted relatively easily, and so even the interpretation part can be gotten wrong by a lot of people. So when I talk about phylogenetic trees, I mean something like this. If we break down the individual parts of it, so we have in the filled circles what are called the tips of the phylogenetic tree. They represent genetic sequences that someone went and collected from an organism, and then using phylogenetic methods based on the patterns of mutation sharing between the samples, we reconstruct the evolutionary history of our samples that are related via what are called internal nodes, which represent the common ancestors. And because this is a hierarchical data visualization technique, we can see, for example, here that samples A and B are more closely related to each other than either is to C, because they share a common ancestor more recently. This particular layout is called a rectangular phylogenetic tree. There are others which I would argue tend to be either as good or substantially worse than this rectangular layout. So if we look at what we can see on a phylogenetic tree in terms of data visualization, we clearly have an x-axis, which usually represents some amount of change happening in the tree on each individual independent branch. This is usually expressed as number of mutations, or using fancier models that can be transformed into actual time. We often don't really care about the history of the samples themselves. We usually care about some process that happens on the phylogeny that can help us disentangle essentially the process. So for example, here we have sampled A and C in state alpha, and our sample B in state beta, and if we infer where the switch from state beta to alpha took place, we see that there's a single transition just at the base of our sample B, rather than the alternative scenario could be, for example, 2 transitions into state alpha on branches C and A independently from state beta. And that's a substantially different story. The other thing to note is that we don't really have access to the y-axis in this phylogeny because we have used up that axis space to make sure that our samples occupy distinct positions, and we use it up to highlight the hierarchical relationships between our samples. And so effectively that particular space is wasted, or at best we can think of it as some proxy for the number of tips that we have in our tree. And also, because this is a hierarchical data visualization technique, it really doesn't matter how you rotate the internal nodes, which is one of the reasons why interpreting phylogenies can be a bit hard. But overall, the point that I'm trying to make here is that phylogenetic trees can contain a lot of information, but fundamentally they are a very low dimensional data visualization technique. And actually, there are examples where people were just very careful about what they tried displaying on a phylogenetic tree. So here, for example, Trevor Bedford and colleagues have used a particular value of a trait that was inferred to shift along the phylogenetic tree itself, and then use that as the y-axis with time being on the x. And because this particular trait value is ever increasing as you go forwards in time, this sort of visualization just works pretty well. But I would argue that if you go with single dimensional visualization techniques or single dimensional data, those lend themselves quite easily to nice visualizations, but in reality are quite difficult to come by, like for example Napoleon's March on Moscow, as visualized by Charles Minard. And even then, as we have these sort of low dimensionality issues when it comes to phylogenies, modern phylogenies actually have hundreds of samples easily. And in fact, what I'm showing you here is just a tiny fraction of a much bigger tree of SARS coronavirus 2 genomes. This particular tree has nearly 100,000 tips, which was actually too big to put into this presentation, and even plotting this caused issues because of the aspect ratio. And even going with a ridiculous aspect ratio, you can see that there's still quite a few artifacts within the tree where the branches sort of clump and you cannot really resolve them very well. And so I would argue that because these sorts of visualizations are quite common in the field, even when they are this big, showing all of the data in the tree is kind of like treating the phylogeny as a very fancy table. Because on top of the information that you're showing to tell a story, you're also including a lot of detail that is technically true, but entirely superfluous. And so I tried to come up with, for this particular presentation, of ways in which we abstract some parts of the phylogenetic tree to make the visualizations much easier. And so the first obvious abstraction where we can sacrifice a bit of something in the tree to highlight other parts is the nested hierarchy itself, i.e. The relationships between our samples. We can also play around with the metrics. So for example, as we saw Trevor Bedford playing around with the space in which the phylogenetic tree was being depicted, we can do the same thing using different metrics. And finally, this is a bit of a cop-out, but fundamentally the reason why people do phylogenies is to highlight some sort of change that happened through time. And so every tree tells a story, and hence it's just an inherent feature of them. So the very first visualization that I'll discuss arguably sacrifices the least of any of these features, and they're called Mueller plots. They're a pretty old visualization technique, but not encountered very often these days, where effectively what we have is the frequency of particular genetic variants through time, and these frequencies are nested such that you can tell what is related to what other things and what their eventual frequency is. And that can be pretty useful, and it's very common in, for example, bacterial phylogenetics. This is my attempt at sort of replicating it. So here what we have is a phylogenetic tree at the top. It is rooted in time, if you like, with samples being collected over time. And at the bottom, we're just tracking a smoothed frequency of some branches that are highlighted in the tree. And for example, no matter how much you stare at the phylogenetic tree itself, I would argue that you would probably not be able to see the vaguely cyclical patterns that happen between the branches colored red and the branches colored blue. So already this type of visualization can reveal features of your tree that would not be apparent otherwise. Another thing that's done very frequently where we can actually start sacrificing bits of the tree is, let's say we have a scenario where we have two groups in our tree, the A's and the B's, and maybe we don't actually care that much about how the A's are related to each other, or how the B's are related to each other, but maybe we want to highlight the fact that A's as a whole and B's as a whole are related, and in fact this is called collapsing clades, i.e. genetic groups, where you simply show where the group began, when the last sample within that group exists, and essentially how many samples exist within the collapsed clade. And this is a very standard approach where you have essentially just sacrificed a couple of nested hierarchy features in the tree for a clearer visualization. Another visualization which I believe also, it clearly sacrifices a bit of the nested hierarchy present in the tree, but I think it can also highlight a lot of the dynamics that would not necessarily be apparent from the tree itself. And in this particular technique, you identify changes from one state to another. So here we have one change from red to blue and another change from blue to red. And what we end up doing is we end up traversing the trees, sort of the subtrees, for as long as we stay in the same state. So for example, here we would traverse this tree because we would ignore the branch that transitions from red into blue. Once we end up in blue, we would traverse these parts of the tree. We would ignore the bit where it goes from blue to red. And finally, we have this small subtree that results from the change from blue to red. And if you then group the subtrees according to color, in this case, not only do you get some better aggregation of your subtrees that give you some indication of how your colors are distributed across the tree, the other nice thing that happens as you traverse the subtrees is that it gives a very natural indication of how long you end up circulating in a particular color here over time. So just to exemplify that, I'll talk very quickly about another coronavirus that I've worked with called MERS coronavirus. So this particular virus primarily circulates in Saudi Arabian and African camels. Occasionally it jumps into humans, but humans are a bad host for this virus, and so the virus doesn't transmit as easily between people, and so that's the reason why when this virus emerged, it caused a huge media uproar in 2012, but did not result in a pandemic like now. And so this is exactly what this particular phylogenetic tree is showing you. It's showing you the branches where we infer the virus to be circulating in camels. We have the branches where the virus is inferred to be circulating in humans. We find a bunch of human clusters where the virus goes into humans but doesn't really jump back out. And even though I, as a phylogeneticist, could get that picture from just looking at this particular tree, if we run the visualization that I was describing earlier, we can really sort of crystallize the idea present within it, where we have essentially uninterrupted evolution of the virus in camels as a single tree, and then multiple very short-lived sort of subtrees that result from jumps of the virus from camels into humans. So here, this particular data visualization technique really shines, and I hope you agree. Finally, there are visualization techniques that we sort of saw before, where we can sacrifice the metric space that we're using in a typical phylogeny and project the tree into a different space. So for example, this is done very frequently in a subfield of phylogenetics called phylogeography, where rather than showing the tree as is in this sort of hierarchical form, you just project all of the events that happen in the tree onto something familiar like a map. So here what I'm showing you is a map of West Africa, the 3 countries that were most affected by Ebola virus during the West African Ebola virus epidemic. And what you're seeing is essentially a tree of over 1,600 Ebola virus genomes, representing around 5% of the known cases. The size of the circles that you see correspond to the number of branches that are present in a particular location, and every migration that we see within the phylogenetic tree is shown as an arcing line that's colored by the date when the event occurred. And so this type of visualization really helps us see the tree in spaces that are far more familiar, but also in this particular case, the data visualization is also very chaotic. It's partially the point of it, but ideally we want something that's a bit more interpretable, and I've tried making this a bit more interpretable using small multiples, but even then, all it really communicates is that there were time periods in West Africa during the epidemic where things got really chaotic, and there were times when things were a bit calmer. And so what I ended up doing for Ebola virus in the first case, and what I did recently for SARS coronavirus 2, is I made an animation where we only really focus on a tiny slice of the tree. So this is roughly what you'll see in the next slide. So just to go over what I'll be showing is, first we have the phylogenetic tree that will appear over time as we animate. It will be colored by the continent where the virus is inferred to be circulating. We'll have just below the tree the number of cases at the time when I was running this animation, and we'll have that Mueller plot-like graph that I showed earlier that's just tracking the frequency of particular groups within the larger phylogeny. And finally, we'll have the migrations that I showed earlier, or similar to what I showed earlier, that will just indicate a virus that's migrating with an infected person between different continents. And we have this Dymaxion projection over here, and also just sort of small insets for the US and Europe, entirely because those are the places that had sufficient data at the country level at the time when I was making this. I will not apologize for using a Dymaxion projection though. So if we begin the animation, we start in sort of late 2019 when the virus emerged in Asia. As time goes on, the virus eventually spread to other places, particularly Europe. And as we head into February, virus migration just really took off as more people were getting infected and more people just took the virus with them as they traveled. The animation gets really chaotic at around this point, again, partially the point. And then what will become very clear right now is that as countries went into lockdown, that really large volume of migration goes down very effectively. Now, one of the points that I'd like to make about using the animation in particular is that even if I had access to the entire phylogenetic tree that contains all of the information that is being shown on the map, I would probably not be able to work out that there's a very concentrated spike in migration at one point in the tree and very little migration in other parts of the tree. And so the animation really helps us intuit all of the information that's present in the tree. And so the tree here is also superfluous. Now, I have discussed data visualizations that abstracted either nested hierarchical aspects of the data or changed the metric space, but I haven't talked about phylogenetic trees that don't have any dynamics in them, and in fact those are possible. So what I'm showing you right now are essentially a phylogenetic tree of all the genetic lineages of SARS coronavirus 2 that have been designated. The width of the triangles indicates how many sequences fall within it, and there's actually a much nicer way of visualizing this where we don't necessarily care much about the dynamic aspects of it, and that's actually a tree map, which is really an underutilized data visualization technique in phylogenetics, which I hope to alleviate in the near future. Finally, I would like to leave you with a data visualization that is quite dense with information, but which I think achieves its purpose even while using a very basic phylogenetic tree. So something that I would argue potentially doesn't need more abstraction, but I am open to suggestions and I would like to hear your thoughts. So this particular phylogenetic tree shows two lineages of Zika virus that were imported into Florida by infected travelers that had spread locally. So in here we have Florida introduction number 1, and here Florida introduction number 2. Importantly, during the study, my colleagues and I were able to show that the virus circulating in Florida, essentially very closely related viruses were circulating both in humans that never traveled to Zika endemic areas, and also the mosquitoes that were caught in Florida around the Miami area. So that's an important aspect to show. We also have a way of saying that both introductions happen independently, because just purely by chance, other people had found viruses circulating in the Caribbean that were each more closely related to the two Florida introductions than either of the Florida introductions were to each other. So that's another important thing. And finally, we also just had violin plots to indicate our estimates for the time when the introduction happened. So if you want, just as a challenge, I'd very much like to hear if you can come up with a more elegant way of showing the couple of features that I highlighted. So the fact that there's two introductions, the fact that we have an intermingling of human and mosquito viruses within each of the introductions, the fact that both introductions are derived from the Caribbean, and the fact that we have some estimates for the timings of some events happening in the tree. With that, I would very much like to thank you for your attention, and just to quickly acknowledge there's a number of people who worked on these projects beside me. Oftentimes I just did the visualizations, so here are some usual suspects and some specific people who helped a lot with particular projects. Thank you very much.
All right. Thank you, Gytis, for that really cool and really, really interesting talk. Phylogenetic trees are a hot mess. So,
Absolutely agree. Thanks for inviting me, by the way.
oh, no problem. And I appreciate you cataloging the ways that they are a hot mess and the way that we can make them better. So there were some questions that were coming up through your talk, but you actually did answer them during it, like how you chose the encodings, like the colors and the sizes and things like that. But I was wondering whether you could build a little bit off of some of the stuff that David mentioned, which is, as you're building these phylogenetic trees and you're looking for different ways of visualizing them and making them more accessible and interpretable to other people, how do those decisions change when you've got, you know, a variety of different people looking at them in your experience with both Ebola and now with you know, SARS and MERS and all that stuff?
Very good question. So I think it's now a bit more accepted to have things that are not strictly just phylogenes. So a lot of the abstractions, for example, we saw in David's talk, you know, the like kernel density estimates and things like that. Yeah, it's not. It's a difficult question, I think.
How did you find like the response to your Ebola viz and even just like those SARS viz that you showed? I felt they were so evocative to really show the problem there. So do you think that something like animation produces that kind of an evocative response and gets people to kind of engage with the visualization more? Or do you think it's just like really tinkering with just that base sort of the first look that you see?
Yeah, so if you look at the phylogeny, as I mentioned, it's very difficult just by looking at a phylogeny to realize what's going on in time. And that's why I compare phylogenetic trees to tables in the first place. But I think once your focus is essentially narrowed to a single point in time, which is what animation is doing, essentially you just pick a time slice through the tree and then you show what's going on. And yeah, I think that is able to really intuitively show in familiar spaces like maps everything that's going on. And I think that's what really hit people hard. And I think it's somewhat it's a bit of a paradox where both the Ebola animation that I've made before and the SARS coronavirus animation now, both of them, they don't really show the data. They just show that everything is a mess. And I think that can be a powerful thing to show. And I have been known to produce visualizations like that.
Thanks, Gytis. All right, with that, why don't we transition to Sidney's talk?
So my name is Sidney, and I am just delighted to be here to talk to you today about kind of a tricky problem, which is, how do biologists actually go about finding meaning and pulling insights out of what is often like large-ish, very messy, very nuanced data, right? So as most of us as data visualization practitioners of some flavor know, data visualization requires being able to really iteratively shift your field of view from one slice of data to the next, and to be able to look at the intersection between variables and kind of build up a mental map of what is in your data and where there might be interesting signals to explore, right? I mean, we end up, whether we're in Jupyter Notebooks or RStudio or another tool like Tableau, we end up kind of iterating through until we have a mental sense of context for a given data set, right? And there are ways that we can capture that context and that iteration, right? So, I mean, small models are a classic, where, either this can be representative of either, the iteration that we're doing on our own within a notebook, or of course we can put them together in small multiples to try and share that sense of context with our audience in a static way, much like Gytis was talking about, how you can have the animations and you can iterate through as an alternative to small multiples. But ultimately, an unavoidable thing is that when we exit the interactive environment where we have really good mental context, and we try and share in a static way, we lose information and we lose some context. And so it makes this process of transferring this mental model of the data that we've built up in our heads through working with the data to others really hard. And this is especially a problem in biology, because while more and more researchers are starting to build both computational skills and domain knowledge in their training, a lot of, like, for many researchers, being able to use both of those skill sets together for their work requires like some sort of a collaboration, right? So often, one person will have really deep computational skills and maybe a little bit of domain knowledge, and then the other person will have, maybe a lot of domain knowledge and like maybe a little bit of computational skills. But basically, as soon as you get big data that is big enough to not fit in Excel neatly, you kind of got to hold things together by duct tape. And, I don't know about you, but in my case, a lot of that duct tape looks something like this email, right? Where, there are then 1,000,005 little iterations back and forth with your collaborators trying to figure out exactly which like facet of the data they're trying to look at and why, so that you can actually appropriately export that in that context. Now, this is frustrating for all participants, and of course we have that information loss that we'd like to avoid. And so, you know, one way that we can do that is try and work ourselves out of a job, at least of this particular part of the job, by trying to empower our collaborators to get hands-on with their own data and to enable them to do the same kind of iteration and faceting that lets us build up a mental picture of the data, but in a way that doesn't require them to code necessarily. So I'll tell you today about two projects that I've worked on that both are interactive explorers with the goal of helping scientists get hands-on with their data. So the first one I'll tell you about is called Cell by Gene. And it is primarily used in what's called single cell biology. We'll get there, don't worry. But under the hood is fairly generalizable to large matrix form data sets in general. And then the second one that I'll talk to you about is very closely to our last presentation by Gytis. We actually were lab mates in grad school. And it's called NextStrain, and it's used for what's called genomic epidemiology. And it's fairly domain specific and works with typically smaller data sets. So before we get started talking about Cell by Gene, so I want to acknowledge that this is a huge team effort, and I especially want to highlight Colin Meagle's contributions here, and that he got the project started and was the lead on a lot of the information design here, as well as the rest of the original team. So Bruce Martin and Charlotte Weaver are software engineers, and of course, like none of this would be possible without actually a very large crew of people contributing insights and ideas and feedback. So when we say that it's an interactive data explorer for single cell transcriptomics, that sounds like a mouthful. So let's talk about coffee instead. So just like a latte or a cappuccino or an Americano are all made of the same ingredients, just in different ratios that determine their identity, the same is actually true of your cells, right? So every cell in the human body has the exact same genome. What makes a heart cell different from a muscle cell, different from a skin cell, is which parts of that genome they're using in what quantity at any given time, right? And it turns out that just with those ratios, we can track as cells become different from each other, all the way from a stem cell to many different kinds of red blood cells and many different kinds of white blood cells, right? And within the last, I'd say probably 5 years or so, we have developed methods that can take a single cell and can look inside of that cell and measure the extent that they are using any given gene at any one time. There we are. And so that data looks like a matrix of cells as rows or observations and genes as features or columns. And then we maybe have some metadata that goes alongside it. So then how do we make sense of this data, right? Like just staring at a giant block of matrix values is not super helpful. So let's dive into the actual interface. So this is a lot, but that's intentional, right? So what we're looking at here is a data set where researchers from the Bush lab took immune cells from both healthy and SARS-CoV-2 infected patients, and basically asked which genes were being expressed in one group more than the other, right? So here in the primary visualization, we have, each dot represents one cell, and we're viewing them projected into a low dimensional space. So looking at 2 dimensions of a UMAP embedding. So as a biologist, one of the first things I might want to do is see, to what extent the course level structure in my data is actually being driven by which donor it came from, right? So this can help me understand, whether, the signal that I think I'm looking at is actually just because of which donor a cell came from versus actual true biology underlying it, right? And then, if I want to dig a little bit deeper, I might go ahead and look at cell types, right? So importantly, we get the marginal distribution for free here, right? Where we're looking at, for each cell type, what proportion came from which donor, right? But here, let's say that I only care about monocytes, right? So what we can do is we can then go ahead and we can do this kind of cross-filtering and faceting that we were talking about, where I can say, I only want the monocytes, and I want to then compare those from healthy controls, and we'll save those as one set, or then I want to compare those directly to people that were suffering from COVID. And we'll save those two subsets, and then we can actually compute what's called differential expression. It's really just a t-test, right? And then we can see, which genes are more highly expressed in this particular cell type in people who had COVID-19 versus healthy controls. And here I can see the marginal distribution of these genes broken down by category. So this is a lot, right? There's a lot in this interface. But the most important thing is that it is information dense, right? There's no wasted white space. It is uncluttered, but it's very information dense. I'm not having to go dig for different features or metadata variables in my data set, right? It is really powerful in its ability to cross-filter and select different subsets of my data and to compare them to each other. And you'll notice here that even though, under the hood, this thing works with basically any matrix data set, like there's not a lot of magic here, but it has been tailored to biologists' experience, right? So, we're talking about cells here, we're talking about genes over here. And most importantly, when we implement a little bit of computation, there's not a lot in this interface, but it is using the algorithms that biologists expect when they're working with this kind of data, right? So I want to compare this to a very different interface, and then we'll bring it all together in terms of what shared patterns we see and what shared challenges there are. So NextStrain, again, is a very different kind of project, looking at genomic epidemiology. And again, first and foremost, I want to point out that this project was started really by Richard Mayer and Trevor Bedford. You'll recognize Colin from the last slide. We've worked together for a long time now. And then recently, James Hadfield and Emma Hodcroft have really led the project over the last few months. So it turns out when we're thinking about tracking a virus across the world or even within a community, there's this really convenient fact, which is that many viruses both evolve and spread on really similar timescales, right? So here, if each of these circles is an infected case, we can see that as it transmits from one person to the next, roughly every other transmission, we see some sort of a mutation occur in the viral genome. This is totally normal and not at all cause for alarm, but it does basically assemble into a barcode, right? Because as soon as that mutation occurs, it's then passed down to all subsequent cases in the same transmission chain. So what this means is that if we are looking at 5 patients and we take a sample of their snot, and we then sequence the virus that we find in that snot and compare them to each other, we can immediately see where here the genome is laid out in a linear way, and then we're looking at mutations indicated by the little circles. We can immediately see that A and B are identical to each other. They have the same sets of mutations. And so we can say that those two are most closely related to each other compared to everything else. And then we can see that C is their closest neighbor because it shares 2 of those mutations, even though it's missing one and has one unique, right? So we can build up this idea of nested relatedness that Gytis was talking about earlier. And we can visualize that in a phylogeny. And here shared mutations indicate that there is a shared transmission history, right? That these two viruses were most likely circulating through a very close transmission network. So let's take a look at how we actually wrap our heads around this data, because as Gytis just showed us, this can be really hard because trees are hard, right? So this is the next train interface. And here what we're looking at is we're looking at 3,500 genomes, primarily from North America, but with a few from other places in the world thrown in for context. And one neat thing we can do is we can actually go ahead and play basically the same animation that was inspired actually by Gytis's original animation for Ebola. And we can see that as the pandemic unfolds, we see a huge outbreak in Asia, leads to an outbreak in Europe, and then we see a lot of transmission throughout Europe. And here, if we scroll in, or if we zoom in to the US in the later parts of the outbreak, we actually start to see that not only is there a lot of inbound transmission from elsewhere, there also starts to be a huge amount of transmission between different states, right? So this highlights the fact that, or it helps us understand the fact that the reason that one of the many reasons the US can't quite get a handle on this outbreak is because you can't just squash it in one place. You have to squash it all over the place at once in order to prevent another outbreak just cropping up again because it gets imported from somewhere else in the country, right? So here, so if we zoom out to the whole picture, one thing we can do is we can then do this same kind of faceting, right? Where we can say, okay, let's look at the whole picture and let's just look at clade, right? And we're curious, which clades have been circulating around the world. There we go, Zoom was in the way. And we can see that, here we can turn on another little plot here. So this is the same kind of Mueller plot that Gytis was showing earlier. We can see how each clade increases or decreases in frequency over time. We can also take a mutation of interest. So this is the mutation you probably heard about in the news, looking at site 614 in the spike protein. And we do see this really interesting thing where, and we can color each tip in the tree by this. We can also color the map. And what we see is that, originally we had the D allele in that site. And then once it transitioned into the G allele, it then really swept across the world, right? And there are lots of possible explanations for this that we won't go into now. But it does allow us to do that kind of overlay of all of our different variables. So what this looks like on a more local scale, for example, and this is fictionalized data but pulled from an amalgamation of real case studies, is we can think about things, if we're looking at just our community, right? So, just the 50 or so samples that were sequenced in our city or our town, for example. We can ask questions like tracking introductions, so whether a bunch of cases are all travel related or whether there's community circulation. We can look at ambiguous contact tracing, right? So, if we had an outbreak in a workplace, is this new infection that was picked up in the same workplace a month later, is that the result of continued circulation within that workplace, or is it just coincident, right? Or, we might want to know whether we're seeing transmission from one town to the next, and whether two different outbreaks that we think are separate actually are, right? So these are two really different explorers, but they have some shared patterns, right? So the first is that both interfaces are information dense. The second is that the state of the data set or the state of the subselection or facet of the data that you're looking at is consistent in every pane of the application, right? So we're not asking people to keep track of exactly what selections they've made, because they're consistent across, which enables a better sense of context, right? And of course, you have this kind of iterative cross-sectioning and revisualization of data that lets people really get hands-on with it. So they also have some shared challenges, right? Where, as Gytis very elegantly articulated earlier, there sometimes comes up a trade-off between what is scientific precision versus interpretability. So believe it or not, these are the exact same tree that we were looking at earlier. They're both very valid representations of the same tree. But here the x-axis is shown in mutations and the y-axis is shown in time. And we can pretty validly infer the time, like where on the x-axis we should place each internal node or each sequence. But there is variability around this, which is why we see such major differences between mutations and time. But if I'm an epidemiologist and I just want to know when the virus started circulating in a given place, I'm going to want time on the x-axis rather than necessarily mutations. And so there is a trade-off there to some degree, and both applications have this trade-off. Another shared challenge is the idea of generalizability versus tailored use cases. So cell by gene is relatively general, but again, like we talked about, there are aspects of the interface that have been carefully tailored to what biologists expect and to their existing mental models to help it feel intuitive and relevant to their work. Similarly, with NextStrain, they've tried to go the opposite direction where instead of just looking at viral genomes, which are very small, they're also starting to accommodate bacterial genomes, which are very large and require some different methods that generate the tree. So here we're looking at a tree of tuberculosis. And notably, when we lay the TB genome out on the x-axis, it's just absolutely massive, right? And so there are challenges here going from, what was originally a specific use case for viruses to a broader use case that also accommodates bacteria. And then the last one that I'll talk about is this idea of, where is the boundary between data exploration and formal analysis, right? So with data exploration, like for example, in the cell by gene interface, you can make any arbitrary selection that you want. And then you can run a t-test to compare to any other selection that you want. And that is not something that I would want to show up in a scientific paper. But it is really useful for helping people kind of wrap their heads around a data set overall, right? Similarly, with NextStrain, we can infer with reasonable confidence where the most likely transmissions are coming from and where they're landing. But again, this is not something that I would consider robust enough for like a scientific paper. And so thinking about, how to communicate uncertainty and especially how to communicate the strengths and limitations of the underlying methodology while keeping the interface really intuitive is a challenge across the board. So I'll leave you with a couple of takeaways. The first is that making meaning out of your data requires kind of this iterative shifting of your field of vision into a data set. Interactive explorers can help empower all researchers, regardless of their computational background, to get really hands-on with their data. And that, really to make these things effective, it requires very close partnerships between scientists and domain experts and information designers and engineers. So again, special thanks to Colin Meagle, who I've collaborated with closely on both of these projects. And huge thanks to the entire team that has worked collaboratively on both of these projects, both on cell by gene at CZI and NextStrain at both the Hutch and Switzerland. And with that, I will take any questions.
Okay, great. Thank you for that really illuminating talk, Sidney. We have some questions for you and that have come in, and then there's also been some questions that have come up over the course of the presentations today. And so I'm also going to pose some questions for the other speakers that have come up as well. And then I'll ask you all to have one final comment before we wrap up for this morning's session. So Sidney, some of the questions from you are, well, here's a really nice one. What do you think the challenge of precision versus interpretability is when there is an actual limitation on phylogenetic trees? So, for example, there's only so many pixels on the screen, right? At some point, you're going to sacrifice both precision and interpretability. What do you think that main challenge is? Yeah. And when we encounter it, what do you think we should prioritize?
You know, I think this, like all data visualizations, data viz questions, it's really about who you're trying to serve with this visualization, right? If you are trying to serve really deep domain experts who solidly understand the methodology, et cetera, then certainly, like in my own work, I want to prioritize scientific precision, right? Because I'm wanting to dive very carefully and very deeply into this data. But when I am working one-on-one with epidemiologists in California, like departments of public health, interpretability, so long as it doesn't lean too far in the direction of like glossing over important nuances, is where I would bias my efforts. Yeah. I realize that was kind of a non-answer, but
no, I think that it's really valuable to know that there is a trade-off, and then that the VIS imposes its own limitations in that trade-off as well. I have one more brief question, and then I'd like to open it up to everybody that spoke this morning. So the last question, for the tools that you presented, what's the quality control like? How much upfront work does anybody have to do to use these sorts of systems?
So both of these systems have some very lightweight quality control in the sense that it expects data in a certain format, and it will error if there are invalid, like just flat out invalid values in the data set. That said, at some point, you know, we have tried many different ways of thinking about, you know, how to really validate incoming data and how to, like, you know, ensure that it is on a log versus linear scale and all these sorts of things. But at the end of the day, like, you can't imagine all of the possible kinds of data that someone might try and shoehorn into your interface.
Yeah.
And, you know, providing really clear tutorials and really clear guidelines, and, you know, showing warnings when we think there's something and erroring if something's definitely broken. But at the end of the day, you do have to kind of trust your user, and that they either they know what they're doing, or they at least have an understanding of what the tool's limitations are and what, you know, really constitutes a valid data set for this thing, through things like documentation, little videos, etc.
Thank you. That's a great reminder of the importance of all the ecosystem that goes around these sorts of tools. All right.
Oh, yeah.
So we're now going to just open up. I've got just some final questions. We are at our break time, so I'll try to keep it brief. Gytis, there was one question that came up for you. Our live stream is a little bit delayed from what we're seeing in our call right now. And so the question the question posed for you, Gytis, is how responsive are evolutionary biologists to having their trees updated? And so, like, something changes. Do they like that? Are they sort of disoriented by it? Are they accepting of it?
I think usually I haven't really received any criticism of anything that I've shown in my papers. So I presume the reviewers, few as they are, see the benefit of just seeing some crystallized form of the visualization that really goes well with the text. So I guess in that sense, I was exaggerating things a tiny bit.
And another part of that question is, have you tested how much of an understanding people get from some of the visualizations that you've shown them? So again, you know, do you find that with either animating or Mueller plot or many of the other variations that you showed in that design space that that some work better than others? Or is it one of those things where like, it depends?
Yeah, I think everyone would agree that that question would be best answered by you, probably, right? Because that is your research. And yeah, I mean, like fundamentally, that is, the actual trying to understand how much people get out of visualization, that is entirely lacking. And I don't know, do you have a stronger opinion or
My opinion is I find that people really hate phylogenetic trees sometimes because they're kind of like, I don't know where to orient myself and what's the clinical action? And then you get other people that are like, but I need the tree. So part of it is trying to almost have the right set of context to understand, which kind of person you're dealing with. So it's a terrible, it depends question. And then we can test to figure out what is super bad, but that's not much more than that, at least not right now. It's still like really early days, I think.
But you would agree that it's basically a table, like
yes.
the full phylogeny.
Yeah. Okay, so before I get into too much trouble, I have one last question for all of you, which is you've shown really amazing work on how you're visualizing all of this data coming in during a pandemic, right? When there's a lot of unknowns. So could you take maybe like a minute or so and tell me what you think the biggest challenge has been and where the research community should really focus its effort? David, could I start with you?
Yeah, sure. It's a good question. I mean, I think there's still globally really fundamental necessity for base data level linkage. We've got these wonderful, beautiful ways of visualizing information, but actually getting linked clinical patient information in systems and in countries where the technology is not quite present in certain formats. We've had a very high profile example in the UK where Excel was used. I'm sure you may have all seen this on the news, right?
Yes.
This is
yes.
a fairly fundamental data mistake that happens. It happens, and you can't blame the individual, but using columns rather than rows. But bringing these kind of high level data visualizations together with the base level data that can inform what they actually mean, I think there's a huge gap there to do that in real time seamlessly. I find myself lucky enough to be working in the field of genomic sequencing, but it's very much not used everywhere, and it's a high income kind of country technology that isn't, look at the use in New Zealand. They've been amazing because they've had low cases and they've managed the response to the pandemic incredibly well. But I think there's a real fundamental issue around how do we bring the base level information together so that we can inform these kind of things. I think the visualizations that everybody's shown today are wonderful. And, we're all incredibly excited and enthused by kind of understanding which stakeholders get to have the right information delivered so that they can do their jobs better. I think that's one of the key things. But in order to do that, I think we need some joined up data linkage at the very, very fundamental level.
Thank you,
That's
David.
a very pessimistic approach, but I think it's highlighted it even more within this kind of pandemic that we need to push that agenda a little bit.
Thank you, David. Gytis, I'm going to pick on you next. What do you think the biggest challenge has been in this pandemic?
So I think everyone who's doing sequencing, for example, the UK, where I believe they have the most genome sequenced out of anyone on the planet right now. The problem is that it is not representative. And the way to fix that is to continue to democratize the sequencing technologies that actually allow us to transform the viruses into the sequences that then we can use to inform decisions. And that is really lacking, even in places that you would think of as developed. So right now I am in Lithuania. There's very few sequencing platforms available in Lithuania that would be appropriate for Lithuania's level of development. And so we're having a big outbreak right now because of the elections. And so we really cannot use that sort of information to help with our response. And that is something that if Lithuania needs this, the rest of the world probably needs that even more. So I'm really hoping that continued democratization of sequencing sort of continues as it has for a while.
That's an excellent point. So it's not only data availability and linkage, but democratization so everybody can use the data. Sidney, what are your thoughts?
Yeah, so I'll take kind of a orthogonal view of this, of less about, where the data is coming from, and thinking about, when we're trying to communicate to the public, what kind of data is of the data available, like what kind of visualizations do I think are kind of lacking? And I think the first is consistent messaging of, I was talking to a colleague of mine who is really an expert in scientific communication, and the point that he made is that, as a community, the scientific community as a whole has, each of us has contributed, or tried to contribute helpful viewpoints, but there isn't really like a consistent message that is being like with the same verbiage, the same kind of visualization, the same kind of like consistent thread through that communication. And it's left people feeling a little bit disoriented, right? And so trying to be like consistent and thorough, maybe. And then, keep it simple of, using chart or like encodings that people are familiar with and that make intuitive sense. And then, the big one that I don't see as much of, even though it's incredibly important, is, and also because it's hard to do, is, what is really the impact of people's choices? Like, for example, Gytis showed in his animation this really clear picture that, once you impose shelter in place, the chaos dies down, right? And finding ways to really highlight that sort of thing for people so that they feel that sense of efficacy between the sacrifices that they're making and the societal outcomes. Like one of the best examples I've seen of this comes from actually a gamified, like basically sandbox test pilot thing from Shirley Wu, looking at, you can toggle different behaviors and then you see the different outcomes, et cetera. And again, like helping people really feel like they are in control of that visualization and the outcomes that it implies, I think is really powerful.
That's fantastic. That is a really, really good and important point. And I've certainly hear what you're saying. I felt that way as well throughout the pandemic. All right, so I think this will wrap up our morning session for today. I really want to thank all of our domain experts who took the time this morning to come and speak with us. You can't see it, but I can. There's little claps for you guys on the Discord channel. Thank you so much for sharing your knowledge with our community today. And so we'll wrap up now, and we'll come again in 20 minutes for our second session with our data and our redesign challenge. Thank you so much, everyone.
Thanks.
Digital humanities present great opportunities for testing new visualization approaches and evaluation techniques. However, and given the diffuse character and novelty of the field, it may also be intimidating for novel and senior researchers willing to get started in the discipline. In this paper, we propose a data-driven analysis of visualization for the digital humanities to identify key themes, authors, and relevant publications. So if you want to know more, please read our paper.
We present StratGraphics, an approach for designing data-agnostic and fully reusable visualization templates. StratGraphics follows the inverse workflow that common data-driven systems. Rather than starting from data, designers construct visualization structures without relying on a data schema. Data tables are shaped later from the structure of graphics. We demonstrate the power of the approach through a gallery of visualization examples.
White space surfaces are a novel approach to convey depth in vessel visualizations. The core idea is to shift all additional depth cues away from geometry and to use the usually empty space between the vascular structures. This allows us to display functional parameters on the surface and supporting cues on the background. We will explain how to generate such surfaces and how to use them as a canvas to further enhance depth and shape perception.
this talk, we introduce CallFlow, an interactive visual analysis tool that provides a high-level overview of performance profiles from large-scale scientific applications. CallFlow visualizes the flow of resource of a program execution as a Sankey diagram to detect performance bottlenecks in parallel codes. CallFlow simplifies these calling context trees based on semantic information by constructing supergraphs.
Tableau helps you see the stories in your data. It's designed to help you be smarter, so you can make better decisions faster. Connect to the data you care about. Sort, highlight, drill down, or filter your data in seconds. Add calculations to extend your data. With Tableau, you can keep on asking questions in the data until you discover the root cause. Tableau. Answer questions at the speed of thought.
How do you visualize the nanoscale, 1 billionth of a meter, for the public? As part of a Science Center exhibition, we developed an immersive environment that explains nanoconcepts by allowing users to explore nanoproperties. We reflect on how exploration, the confluence of explanatory and exploratory visualization, can be applied to visualization in public spaces.
Entities and their changing relationships can be modeled more precisely as temporal hypergraphs, but hypergraph models are difficult to explore and refine. By leveraging domain-specific geometric deep learning models and a new multi-level hypergraph visualization, our technique allows for the direct integration of domain knowledge into the machine learning process. The multivariate hypergraph model structure can be analyzed on different abstraction levels. Simultaneously, experts can integrate their domain knowledge on the fly, and then explore the refined machine learning model.
Tableau helps you see the stories in your data. It's designed to help you be smarter, so you can make better decisions faster. Connect to the data you care about. Sort, highlight, drill down, or filter your data in seconds. Add calculations to extend your data. With Tableau, you can keep on asking questions in the data until you discover the root cause. Tableau. Answer questions at the speed of thought.
Attention mechanisms have greatly improved the performance of many language models. Yet with great power comes increased complexity. In this work, we present attention flows, a visualization that let users interpret the language model's decisions and gain insights into the underlying self-attention mechanism. We also support model comparison that helps to fill the gaps between models in different training stages.
While meetings are an essential part of everyday business life, most calendar visualizations are not designed for retrospection. We present a tool consisting of 4 coordinated views that integrate into the user's daily workflow and are designed to promote self-awareness about past scheduling decisions. We discuss the insights gained from an internal user study and from a field deployment. We also discuss how we strongly inherit from the principles of personal visual analytics despite targeting a professional environment.
We present Lyra 2, a system for designing interactive visualizations by demonstration. To design interactions in Lyra 2, users directly manipulate the visualization canvas. The system interprets demonstrations using heuristics and suggests possible interaction designs. We find that Lyra 2 enables rapid prototyping of an expressive range of interaction designs. We're excited to share this work with you.
Tableau helps you see the stories in your data. It's designed to help you be smarter, so you can make better decisions faster. With Tableau, you can keep on asking questions in the data until you discover the root cause. Share your analysis securely. Your entire organization can access these interactive dashboards from any browser or mobile device to find their own answers. Tableau. Answer questions at the speed of thought.
CarxS is an authoring system which enables rapid creation of zooming-based scatter plot-like visualizations in just tens of lines of JSON. We contribute a declarative grammar for such visualizations, which captures a large design space. By working with multi-node database spatial indexes, CarxS enables interactive browsing of billions of data points. Come listen to our talk to learn more.
How many clusters can you see in these images? With different visual encodings in scatter plots, our perception of cluster count changes. We developed models that consider how visual density influences cluster perception. Further, we demonstrate using a threshold plot to optimize the saliency of clusters.
As commonly known, dimensionality reduction techniques and their interpretations are complex, biased, and uncertain. In drug design, this highly complicates the search for similar and new chemical compounds. To overcome this issue, Canva brings comparison of different molecular descriptors and properties into one tool, featuring planar, 3D, and table views for evaluating the trustworthiness of high-dimensional data projection.
Deep neural networks are vulnerable to maliciously generated adversarial examples. This brings high risk in applying these networks to safety-critical applications. We developed a visual analytics approach to explain the root cause of such wrong predictions. Our contribution contains a constrained datapath extraction method, a river-based visualization, and a contribution analysis method.
We asked participants to recreate bar graphs. When the bar was alone, we saw a different pattern of error than when the bar was presented with context in a stacked bar graph. We find that absolute error increased for higher bars when they were presented alone. We also found that there was a bias in the responses such that they were repulsed from an implicit 50% mark. We found a magic number, 10%, to predict error. Participants are usually within 10% of the height of the bar.
If machine learning were like education, we would like to test what concepts our student, the model, has learned. Has it learned the concept of object rotation? Does additional text help with object recognition? We need a methodology and platform for conducting such tests. In this paper, we present a novel visual analytics tool that enables hypothesis-based evaluation of machine learned models.
String graphs are variants of stack graphs with curve-based line, and the main factor affecting its readability is the sign allurance through a perceptual inconsistency of the orthogonal and vertical direction. Aiming at reducing its impacts, we revisited the baseline formulation and proposed the concept of composition to help the string graph layout optimization. The result shows that our algorithms perform better than the others.
VisFCAC is a visual support system for the development of visualization algorithms. It has live compilation, automatic version control, predefined interactions, and tools for visual parameter analysis. By displaying the complete history of the algorithm, users get insights into the correlation between source code changes and visual differences.
We propose a new semi-automatic method that uses topological features to guide users in tracing neurons
and integrate this method within a virtual reality framework. We use the More-Smell complex to find a set of candidate neuron arcs. The candidate arcs are integrated into a VR neuron tracing system and exposed through our More-Smell complex guided semi-automatic tracing tool. The topological ridge graph underlying our MSC guided tool is robust against gaps in the signal.
In our paper, we address the problem of navigating complex multiscale and dense environments, such as these molecular models. We present a technique for browsing a model by clicking on textual labels, which we call hyperlabels. This allows the user to intuitively navigate the hierarchical organization of the model. For more details, read our paper or watch the talk.
Morse complexes have shown a great utility in understanding the topology of complex scientific datasets. The noise in the scalar field, however, can significantly distort the Morse complex topology. Hence, we study the extraction of Morse complexes for ensembles of noisy scalar fields through their uncertainty visualization using our proposed statistical summary maps.
Visualizing class separations is used in applications such as classification and clustering. However, many dimension reduction techniques are limited due to the issues of separability and interpretability. We propose a visual analytics framework to support the exploration of nonlinear complex separation structures with the power of locally linear separations.
We have seen many visualizations for tree data structures, but when showing changes in trees over time, some focus on displaying hierarchy, while others highlight their changes. With split streams, we combine two approaches and their individual benefits. We evaluated our method in a user study and consider it a general-purpose method for the visualization of dynamic hierarchies. Our JavaScript library is openly available.
Progressive visual analytics provides intermediate results in the middle of computation to address a long computational delay. However, such intermediate results are uncertain, so findings from these results can turn out to be incorrect for the entire data. In progressive visual analytics with safeguards, we suggest to risk the uncertainty, but we allow humans to leave safeguards every time they risk the uncertainty.
Linking and brushing is very useful for interactive visual data exploration. Data-driven brushing tools are getting popular recently and achieve good results, for example the Mahanoda-based brush and the CN-based brush. However, these brushing tools are optimized by the data from a large number of users, which are not suitable for everyone. In this paper, we introduce an adaptive brush model which takes the user in a loop to improve the brushing accuracy.
A testing environment for continuous color maps. Many other fields in the computer science do it, we should do it too. With our work, we introduce the approach of using test functions as a standard evaluation method. We present a test suite for continuous color maps implemented in the CCC tool. Adapt the test to your requirements at the interactive testing section and observe them at the testing evaluation section.
everybody, and welcome back to our second session of the BioVis Challenges Workshop at VIS. So we are now at the second portion of our presentations. We are going to be talking about our data and our redesign challenges. And just in a moment to kick this off will be Hilary Coon. And just after the presentation of these challenges, we are going to have our capstone presentation by Niels Gallenborg. So I want to take a moment to introduce Hilary. Hilary provided the data set, or Hilary's team provided an anonymized version of the data set that was used in the data challenge. She is a researcher at the University of Utah School of Medicine in the Institute of Behavioral Genetics, or at the University of Colorado. Her work focuses on the study of risks and suicide, and she has carried out this work by collecting over 7,000 DNA samples on suicide deaths over 2 decades. And she has established this amazing database called the Utah Population Database, or linked to the Utah Population Database, which includes statewide resources that have extensive genealogical records, demographics, and environmental data for more than 11 million individuals. She's going to talk to us today about some of the work that she has been doing on trying to understand suicide risk, genetic suicide risk, and some of the way that she has collaborated with visualization researchers to address these problems. So I am really pleased to welcome Hilary Coon to give the data challenge presentation.
Thanks very much for allowing me to speak. It's really a pleasure, and this is not my usual audience, but really appreciate being able to talk to you today. This has been some terrific work that has been in collaboration with some of my colleagues here at Utah. So I want to talk about this visualization of some of the data in our very complicated project. I don't have any conflicts of interest to disclose. And since I'm going to be talking about suicide, because that's the main focus of our work, I just want to make sure I put this slide up here first. I am going to be talking about this, and I want you to be aware of resources of prevention and also research resources. If you or anyone you know is in crisis, there are many ways to seek out help, and these are some of those ways. Thanks very much for allowing me to speak. Thanks very much for allowing me to speak. It's really a pleasure, and this is, thanks very much for allowing me to speak. It's really a pleasure, and this is not my usual audience, but really appreciate being able to talk to you today. This has been some terrific work that has been in collaboration with some of my colleagues here at Utah. So I want to talk about this visualization of some of the data in our very complicated project. I don't have any conflicts of interest to disclose. And since I'm going to be talking about suicide, because that's the main focus of our work, I just want to make sure I put this slide up here first. I am going to be talking about this, and I want you to be aware of resources of prevention and also research resources. If you or anyone you know is in crisis, there are many ways to seek out help, and these are some of those ways. Okay, so just to tell you what I want to tell you, this is the overview. I want to tell you a little bit about our research program. Our main focus is discovery of risk factors leading to suicide death. And we really had a very specific need for data visualization in one aspect of this study, which has turned out to be a very flexible program. It's been a wonderful tool. So I'll talk to you a bit about the project in general to really motivate that need. And then about the development of this software tool, which is called Lineage. This was in collaboration with Alex Lex's lab here at the University of Utah Scientific Computing Institute. This tool is really very data-driven, it's multivariate, very interactive, very flexible. And then I'm going to give you a couple of examples of uses of that tool, and then talk to you about a few of the next steps that we're using to modify the tool to do some additional things. So it's probably no surprise to you that suicide's a major public health crisis. It's right now the 10th leading cause of death in the US. And this is actually it's number 2 or number 3 if you go down in age to lower age groups. Over 800,000 people die by suicide every year worldwide. That's from the World Health Organization. And suicide attempt is actually much more common than suicide death. For every death, there's about 25 to 30 times the number of attempts. Just to show you a little bit of data from what we're doing in Utah, this is also a rising health crisis. Suicide deaths have increased over 30% in the US since the late 1990s, and in the same time period, they've really increased even more dramatically here in Utah. So we have some very interesting ways of looking at risk factors for suicide. And one of the things that has been a big focus for us here in Utah is sort of thinking about familial risk. The family tree that I've shown you here in this slide is actually not from Utah. This is from a much older, very seminal study done by Janice Egeland in the Old Order Amish isolated community. She did some fascinating work in that community looking at people with major depressive disorder and bipolar disorder. And she studied many of these very extended families with people with these affective diagnoses. And what she found was quite interesting, which was that out of the many families she studied, there were only 4 of them actually had individuals who had died by suicide. And from this, she concluded that it looked as though, number one, suicide risk looked to be familial. And number 2, it really looked like there was something about suicide death that was somewhat specific, that it wasn't just anyone with a diagnosis of depression or a bipolar disorder that would have this risk, but it was quite specific. We have some initial additional evidence from other published studies that convince us that genetics is probably important. It's not just those familial studies. The familial studies, people might be also sharing other environmental familial risk factors. So we have to convince ourselves that there might be some genetic etiology underlying suicide risk from some other kinds of designs. One of these is kind of a natural, interesting human experiment looking at twin studies. So you can look at identical twins who share all of their genes and compare those to fraternal twins who share half their genes on average, just like full siblings. And to the extent that a trait is more concordant in the identical twins versus the fraternal twins, you can make some conclusions about the genetic contribution to that trait. Well, for suicide death, we can see in the data here, this is aggregated data across the literature, that the identical twins are much more likely to be concordant for suicide death than the fraternal twins. Additionally, we can look at adoption studies where if we look at an individual who was adopted, who committed suicide, and looked at their biological relatives with whom they share only the genetic risk factors, and then compare that to their adopting relatives who share only their familial environmental risk factors, we again see this evidence that there looks to be a biological contribution to this particular trait. So if you look at the combined literature, it looks like evidence for genetic contribution to suicide deaths about 50%. It's quite strong. It's worth saying that we really understand that suicide risk is very complex. What we really believe is going on here is that individuals may have a biological predisposition to be more vulnerable to the many types of environmental stressors that could predispose them to increased risk. So in our work, we're looking at both genetic factors, but then also trying to take into account many of these environmental risk factors. Well, why would we want to do this in Utah? So right now, Utah is in sort of the dubious position of being at the kind of the top of the states for suicide risk. So it's right now in the 6th position for suicide risk. And suicide right now, it's a leading cause of death for people under age 25 in Utah. We also have some really amazing resources. We have a long-term collaboration with our medical examiner's office. And through this collaboration, we have 7,000 cases of suicide death with DNA, where we've collected DNA. This grows by about 700 cases a year. We also have something called the Utah Population Database, which is a very large statewide database that has medical records data, demographics data, and very importantly, genealogical records that go back to the 1800s and sometimes even 1700s. So we can use that data to look for very large, high-risk families where we see more cases of suicide death than we would expect by chance. And we do this by using a statistic at the familial level that's essentially a familial relative risk number. And doing this, we linked that giant database of suicide death data that we got from the medical examiner with the genealogical data that we get from the Utah Population Database. We selected some very high-risk families, and we did some genotyping. The distant relatedness in these families means that these individuals really don't share very much in the way of environmental shared family risk factors. They're really high risk mainly because of genetic risk that they may share from their common ancestors. And really the chance of finding genetic risk in these families is truly magnified through the familial repetition of these individuals. So it's a really powerful statistical design for looking for genetic risk. Well, we took these families and we selected the ones that were the largest and had DNA on the most number of cases. We used a method that was developed by another one of our colleagues in the genetic epidemiology department, Nikki Camp and Rosalie Waller. And with this method, which is essentially a simulation-based method that takes into account family sharing across the genome, we came up with a bunch of different significantly shared regions in these families. These are 13 regions that we were most interested in because they were overlapping across more than one high-risk family. And you can see there's a lot of them. And we needed to think about, well, okay, what genes are in these regions? And what do these individuals who died by suicide, what are their characteristics, the ones who share these regions? This ended up being a very big problem. The significant regions included over 100 genes. Many of these regions actually had multiple genes. And we know that the risk model that we're looking at, it needs to be one risk variant that is really driving that significance. Well, what gene is it in? How do we prioritize that? We could look at what do these genes do, and that was one obvious choice. We did find several genes here that actually had prior evidence for suicide risk, and these were definitely on the top of the list to look at. But there are a lot of genes in these regions, and many of them may be very interesting. 93 other genes, they have many functions. Some of them already had evidence for being involved in psychiatric disorders, in neurodegeneration, in neuronal development, in neuronal function. These are fascinating. We were also interested in genes that might have influence on immune dysfunction and inflammation as being predisposing for risk. So how do we do this? We've got a big problem here. We have individuals who died by suicide. We do have this familial aggregating evidence. We have a bunch of genes we're interested in. They do lots of interesting things. We've got to prioritize which genes to study because that's expensive. We need to be able to maybe select additional cases within our large family cohort, but then also unrelated individuals to look at for follow-up studies. We need to be able to select additional high-risk families to try to extend these results. Okay, so that's obviously we've got a big problem and we need visualization. The amount of demographic and diagnostic data that we have on these people is also quite overwhelming. And most of these people have essentially lifetime electronic health records. So this is a lot of data. We tried to use some of the existing pedigree visualization tools out there, and we found that this was extremely frustrating. So these tools worked very poorly for really large families. There are tools out there, but they tend to be developed for much smaller family structures. We would try to import some of these families into some of the existing tools and end up with just a complete mess that was impossible to visualize. We also realized that some of our data had some errors in the pedigree structure, and this made the importing into the existing tools really challenging. We also found that there's a very cumbersome way of trying to visualize all of the diagnostic data and the different types of both quantitative and qualitative data that we had on these individuals. So I was very fortunate to be able to collaborate with my colleague Alex Lex and Carolina Nobre in the University of Utah Scientific Computing Institute. And we had this amazing interaction with them to develop this tool. So some of the highlights of this development, we had some interactive brainstorming sessions with them where sometimes as a group of scientists, we didn't even really know what we wanted in terms of being able to visualize. And I remember these sessions being a lot of fun where it would be like, well, we want it to, we want to just be able to put in a different set of characteristics and sort by that set. And then we want to be able to look across 100 different families and see which ones have the highest average number of people with depression diagnosis, or sort by the lowest average age at death, things like this. And it was a fun process because we didn't even really know that we might be able to do some of these visualization techniques. We were impressed to get a very quick access to working prototype, and then again, keep working with the development team to sort of tweak things and refine it so that it worked at the best for our project. And we also had issues about data security. We had to be able to have this be a tool that could be used by people that were not necessarily all that computer savvy. We had to ask them to make the tool be flexible and to be able to add different features to it if that was going to be possible. So let me show you some of this. So this is the picture on this slide is the old-fashioned static tool, but I just wanted to start with this. This is one of our big extended families. The folks at the couple at the top there, so circle is female, square is male. This is a very traditional way of looking at a family tree. People who are shaded in are suicide deaths. The ones with the red arrows are people who actually share a significant genome segment on chromosome 2. This was a family that was really of interest to us because the gene in this segment, there's only one gene in this segment, which was nice. That gene was a neurexin gene. It already has evidence for suicide risk, so it was high priority. And we wanted to know, number one, what's different about the cases in this family at the family level? What do the people who actually share this genomic region within this family look like? We needed to do this to target maybe additional people in the family to do molecular work on, and then also to prioritize other follow-up cases to study. So now this is what the family looks like in lineage. And this is, it's very small. I'll give you a little bit more zoomed in view of this on the next slide. But this is way better for us for being able to sort, prioritize, and look at characteristics of these cases. So the folks I showed you on the previous slide who share this genomic region are highlighted in gold on this particular visualization. We can take these individuals and sort by any attribute in our data set. So again, we have hundreds of attributes that are demographics, co-occurring diagnoses, different types of even molecular data that we have generated on these individuals. And we can sort based on other types of data. Over here on the left-hand side of the slide is a list of the actual families. So we can even choose an attribute and sort the families by that attribute so that if we could see a particular characteristic that was really important to us, then we could sort other families based on that characteristic and think, okay, now I want to look at this other family, see what genomic regions it's shared, or see whether or not maybe this is a good family for doing more molecular work to follow up this particular region. Okay, so this zooms in a little bit on just part of that screen that I showed you in the previous slide to show you a little bit more about what lineage can do for us. So in this particular view, you can see something called lab ID. This would be an indication that we had actually collected DNA on that case. This would be a kind of just a nice way to show us individuals who might be available for more molecular work. It would also, in additional families that we might want to target, show us, okay, is this family actually going to have enough individuals to be informative for our familial genetic design, because we need at least 3 individuals with DNA. They need to have separation by a number of generations in order to give us the statistical power to see the effects that we're looking for. We also have demographic attributes. So we have age at death, we have year of death, and this is important because the more recent deaths have much more deep electronic health records data, so they're more informative for us. The overall, there's a little distribution up here of the overall age at death for the whole family. So it's a possibility that a family could have a familial effect of maybe more young age at death, and this could be genetically meaningful. In this particular example, we have just 3 clinical attributes shown: bipolar disorder, personality disorders, depression. We could have displayed many other possibilities here, other psychiatric diagnoses, other medical diagnoses, medication data. We have something called polygenic risk scores, which are sort of background risk of different types of traits or diagnoses. We actually even now have exposure data and geocoded location within the state of where you were at time of death. So all of these things could be also visualized. So I did mention that we can look at other families. One of the things that we did with this particular project is to think about, at the molecular level, we're looking at this gene, orexin. And we can look at gene pathway analyses using different tools and understand that there are many genes that actually interact with orexin. And we can look at our data from our initial familial analysis and look to see whether or not there are any other families that have sharing in regions with genes that interact with orexin. So these would maybe be families that would have similar genetic etiology on the level of the gene pathway. And lo and behold, we did find another family that has shared a significant region for a gene called LRRTM3. This is a gene that regulates orexin in the synapse. And this then is a showing of a different family, this 2723251. And when we looked at the attributes within these 2 families together, we could see a really unusual association with cases who were sharing these 2 regions having personality disorders. And personality disorders are not actually all that common in the electronic health record data that we have. Something like depressive disorders can be way more common. So being able to visualize something that is a little bit more rare like this and seeing it occurring in a number of the orexin and LRRMT3 sharers was unusual. And we used this information in order to be able to select some follow-up cases when we're looking at actual variants, risk variants in these regions. And also then to prioritize new families for more molecular work. We also are all the time collecting new individuals with DNA who died by suicide. And it turns out when we relink cases into some of these genealogical structures, we linked in a bunch of new suicide deaths into this very large family, original family, 601, 627. There were 26 more suicides that linked into this family after a couple more years of studying this. We now have many more individuals highlighted in yellow here, and these are new individuals that also share this chromosome 2 region in this family, and that we can use this to refine the genomic region where we're looking for variants, but then also, of course, to refine the characteristics that we might be really interested in looking at within the suicide deaths in this family. So what we find when we look at all of these new individuals is that personality disorders were still important, but then we found a few more things. We looked at these polygenic risk scores, background genetic variation, and we found increased risk for intellectual disability, polygenic risk in these individuals, which maybe makes sense given that the orexin gene is implicated in neuronal function. And then we also found some indication of diagnoses having to do with inflammation. These are not necessarily psychiatric diagnoses. This is really of high interest to another colleague of mine who's been doing work showing that short-term exposure to air pollutants, particular air pollutants, is potentially a risk factor, a trigger risk factor for suicide death. And she's been using some of the individuals in this family for some gene by environment interaction studies that she's designing. So those are some fairly specific examples. We're also using lineage for some more general examples. One of these would be really trying to prioritize youth suicide. Younger cases really show very high polygenic risk of suicide death, so they seem to be sort of genetically enriched. And we've been using lineage to sort by family, just using that low average family age at death to select families in that look like they have the most statistical power for additional molecular work. We've also been using lineage to prioritize for a project that's really focused on individuals with bipolar disorder. Bipolar disorder has very high co-occurring risk of suicide death. It's about 10 to 30 times the population rate. And so this prioritization using lineage, we can even refine this further. So we have done some epidemiological and clinical studies that suggest to us that bipolar disorder plus trauma plus post-traumatic stress disorder diagnosis plus anxiety diagnoses are really potentially a very high interest as far as maybe a genetic risk group. So then we can use lineage to go through and select families with constellations of these particular diagnoses. This is incredibly cumbersome to do without the visualization tool. So we've really appreciated having this for prioritizing. This is just to go backwards just a little bit and show you one of the youth suicide families. This family has an average age of death of only about 27 years. Just more than 14 years younger than the average overall cohort mean age at death, which is in the early 40s. So we can do this, and this family ended up being one of the most extreme families for this study, and we've done some molecular work on this family. It looks incredibly genetically informative. There are many interesting shared regions among the individuals in this family. And we found by looking at attributes that substance use and some other aspects of background genetic risk are really important in this particular family. So those are some of the things that we're doing right now. We have additional family subtypes that we're using lineage to look at. These include psychosis, postpartum suicide, autism. These are individuals in our group that are looking at these conditions and have been using the tool. We also have other sources of data that have come online that we can import into the tool. These include prescription data and electronic health records, autopsy toxicology data looking at presence of drugs of use and abuse, looking at evidence of opioid use and misuse and trying to disentangle that issue from suicide death. And then incorporating, I mentioned this briefly, localization of suicides, looking at geocodes. This can be looking at urban versus rural risk factors. It can also be looking at exposure data. We've got an individual now looking at epigenetics, and again, this is trauma and exposures, and importing the epigenetic data into the tool will be very interesting in the future as he generates more of that data. One of the things that we've also thought about doing is altering the tool so that we can input data from suicides that aren't linked to the family, just for more interactive sorting for follow-up cases. Okay, so getting to the end here, I just want to remind you that there are likely hundreds and hundreds of genetic variants that might lead to small increases in suicide risk. These are going to interact in very complicated ways with background polygenic risks, with other complex environmental risks and exposures, and that really no one genetic change could ever cause suicide. So we've got a very big problem in front of us. It's really good to have these visualization tools to help us navigate this very complicated work. And a reminder again about prevention services, just making sure that you stay safe. I'd like to acknowledge that I have lots of wonderful collaborators. The goal on this slide is to make the font so small that nobody can read it. The ones I've highlighted here right in the middle in red are my colleagues, Alexander Lex, Carolina Nobre, who really did the lion's share of the work here in developing this tool and have been just absolutely delightful to work with, and many donors that have contributed to this work. Thanks.
All right, thank you for that great talk, Hilary. I absolutely loved how we transitioned this morning from looking at trees for viruses, now looking at trees for people. We're just waiting for the questions to come in, but I wanted to start by asking something that I think is super relevant to the Viz community, which is Lineage was clearly a really, really helpful tool for your collaboration process. What was it about the way that you worked with the computer scientists that made it so useful? Was there something they did in particular that worked for you to get such a great tool going?
Yeah, sure. So the collaboration was very interactive. We met with the team several times, just sort of brainstorming what types of visualization would be the most useful and how it would work. And I would say we have many people in our group, including clinicians, epidemiologists, and computer science experts, but lots of variety. And many people had different ideas about how they wanted to see the tool work. And the development team were very flexible in this. So there was an initial sort of workshopping almost of how the tool would work, and that was really great. A lot of times we didn't even know what it could possibly do, so that was helpful. And then also just getting to a working prototype really quickly, and having the data, the software team be able to handle pretty ugly, messy data from us, not needing a lot of help with that. It was pretty impressive. And then once we had a prototype, then still a very interactive process to make it really what we wanted.
That's great. I see some people are typing, so I've got one more question here for you. So another question like that came up a lot in our previous session was the challenges of linking data. And I saw towards, some of the extensions that you want to apply Lineage to, as well as even just the current work that you've got where you're linking these genealogies to electronic health records. Can you speak a little bit about the challenge of bringing this data together? Like, what kind of blockers are there for you, and what would be helpful to overcome some of those issues?
Yeah, so definitely this is very complex data, and it's full of missing data, and it's quite messy, and it comes from multiple sources. So that's a challenge. We have a little bit of an advantage in that there isn't like consent for a lot of the data that we're working with, because these are individuals that are deceased. So that's good. Now the family structure data is potentially identifying. So, giving this out to a broader community to look at, we have to deal with those privacy considerations. It's the data structure itself. Actually, this was helpful with the tool, was figuring out where there were errors in the genealogical records and fixing those, and working then with the Utah Population Database to say, hey guys, there's this entirely unhooked branch of the family here. When we've imported it into lineage, we can see what's going on. So that was helpful.
And we've got one question that's come up from the chat here about the workshop. So was the scoping workshop done in any particular form that you found useful? Like, was it just general brainstorming? Were there specific objectives? Was it more of just like a creativity, blue sky free-for-all, or was it a bit more grounded?
Yeah, so Carolina actually led the workshopping and did it in a way that that was kind of novel for us, where we sort of wrote down different attributes that we wanted. We had sort of color-coded ways of saying, okay, these are things that have to do with data. These are things that have to do with clinical interpretation. We sat around in a room and did this for the first couple of times, very sort of personal interaction. And then there were other interactions that were more, okay, here's the tool. This is on your computer screen. You have multiple people looking at it, working with it, and then giving feedback. So it's sort of two levels. The first one being very personal and very sort of, brainstorming in a way that worked out pretty well, and then having it be more computer oriented.
That's fantastic. That's really useful. And for those of you following along in the Discord, Alex Lex has actually posted a link to the paper about what that workshop was like. InfoVis 2018 Creative Workshops publication. I totally encourage folks to check that out and get more details as to what Hilary was talking about today. All right, so thank you so much, Hilary. There's lots of claps in the channel. I will transmit them to you now. We really appreciate your time, and we're now going to move on to the data challenge portion of our discussion. So we released a synthetic data set, and we had some folks that had their take on it, and they submitted their results to the BioVis challenge, and we're going to start off with the first presentation of two. This one is an interactive visualization of multivariate clinical data in a force graph. It is presented by Mingi Ryu, and we are going to load that up now.
Welcome, everyone. My name is Mingi Ryu, and I'm going to present the interactive visualization of multivariate clinical data in force graph. The visualization application consists of 4 core features: the visualization itself, a table view, the selection and filtering based on the dropdowns, and view manipulation based on the buttons, which enables things like toggling between different link visualization, pausing and resuming animation, and selecting visible nodes and zooming based on visible nodes. These are the 4 visualization examples that inspired me to take on this project. The top 2, lineage and next train, are of very complex, interactive, but also intuitive visualization applications. And the bottom 2 are force graph visualization prototypes that were very helpful in getting started. They share a common theme that they're all web-based visualizations. Also, they have interactive components that drive data exploration. And if they have one, they often have intuitive UI and UX for selection and view manipulation. And Lineage specifically has a fantastic tabular visualization for viewing the raw data points itself. The application is built based on 3 core components. First one being the force graph framework. I chose this instead of working directly with the D3 because it provided me with a very easy to use API and also a very extensive features for interactivity. I use semantic UI for the sake of simplicity, but also it provides a very lightweight but feature complete table view, and all other interactions are implemented in pure JavaScript. These JavaScript code works as a binding between the framework's API and semantic UI and HTML event handlers. The takeaways from this project is that these high-level tasks that were given by the challenge can be easily broken down into low-level tasks using the graph task taxonomy. And it is often helpful to do this because then the developers, also the users, could map these low-level tasks to specific interactions. And while they're often ambiguous what these specific low-level task is, or oftentimes a single high-level task can be broken down into many different combinations of low-level tasks, it is once you have a set of low-level tasks, it's easier to communicate what this high-level task consists of. Also, the design choice that I made, which is using force graph framework instead of D3, it represents idea that there are many easy-to-use open source tools out there. And while D3 is often my go-to solution and it is very powerful, it's even more powerful for data manipulation, but oftentimes it's much easier and easy to prototype different applications and visualizations using these open source tools. These are the references. Thank you very much.
All right, Mingi, you can unmute yourself and we will get to one brief question. Okay, great. Thank you for that really great presentation, Mingi. So one of the things that you really looked at was the way that open source tools and available tools allow you to build these kinds of interactions. Could you talk about some particular challenges of designing interactions for a data set like this?
Yeah, so for particularly like these kind of data sets, which is, we could think of it as like these networks with nodes and edges. And oftentimes these data sets really depend on how they're structured and how they're connected, how these communities, like the nodes and edges, are structured. And you can't really design these things ahead. Like you can't come up with interactions first and then apply it to the data set. You usually have to have a iterative approach to working with the data set first and then implement interactions based on what features you want to implement. So those were the challenges, yeah.
So did you find that like the D3 tool in particular allowed you to do that data munging a lot easier than other tools?
Yeah, so D3 is like really powerful in that with Python, you have pandas for data manipulation. With D3, you could do basically most of the things that Panda allows you to in like daily basis, but in that JavaScript frame like ecosystem. So you don't have to like go back and forth with Python and JavaScript.
Okay, that's great. Thank you. These data challenges are really brief. We're going to move on to our next data challenge presentation. That is by Jake Gonzalez. Let me bring up the title here. And he is going to be giving a talk about, or his talk is about, VisFCAC, an interactive family for clinical attribute comparison. So Joy, can you please switch to Jake's talk?
Here's the layout for the visualization created for the BioVis Challenge 2020. This features radial trees in order to display the family structure of each family in the dataset, radial charts to display the clinical attributes of people who have committed suicide, and dot plots to display the distribution amongst all of the families for each clinical attributes using their age. The motivation behind using the radial tree to show the structure of the family was to show how closely related different individuals are amongst different generations. So we can see generation 0 is represented at the center of the radial tree and has branches to each of its offspring. Each family unit is represented by a square for the males and a circle for the females. These shapes have shaded areas in them. This represents the age of the individual. The more shaded area inside the shape means that the person is older. The color of this shaded area represents whether the person is alive, deceased, or has committed suicide. The gray represents that the person has deceased. The bluish green represents that that person is alive, and the black represents that that person has committed suicide. If a person has committed suicide and also has clinical attribute data, a radial chart is shown for that person. This radial chart has different sections colored that correspond with different diseases. These colors can be referenced down at the bottom dot plots to see the name of that clinical attribute. So you can see this person right here has anxiety, non-trauma, and also depression. The motivation behind the dot plots was to show the number of people that were diagnosed with any specific clinical attributes and the distribution of age with people that were diagnosed with that clinical attribute. So you can see each of these diseases has a specific color associated with them. This corresponds with the color that is displayed on the radial chart for individuals that have clinical attribute data. If we mouse over one of the circles that is on this dot plot, we can see the person ID that that circle represents, the diagnosis age, and the family that that person belongs to. The justification behind using the radial tree for displaying the family structure was to show the last common ancestor for individuals who have been diagnosed with similar clinical attributes. So we can see these two individuals have been diagnosed with anxiety, non-trauma, and depression. But it looks like they're on separate branches of the family tree. If we trace back a few generations though, 5 generations for this individual and 6 generations for this, we can see that the family unit that is the last common ancestor was this family unit right here, who had 3 different offspring. In each of these branches, there are individuals who have committed suicide. So we conclude that anybody that is a descendant from this family unit might be at risk of suicide or other clinical disease. One interesting pattern that comes up using this visualization is the correlation between depression and suicide. So we can see in this branch right here, there's an individual that committed suicide who was diagnosed with depression. This person had offspring, one of which also committed suicide and was diagnosed with depression. Similarly, if we look at this branch in the same family, there are 2 individuals in this generation that committed suicide and were diagnosed with depression. One of which who had offspring, one of which who also committed suicide. And another one of these descendant branches, there is another individual that committed suicide. If we look at the dot plots, we can see that depression is the clinical attribute that was diagnosed most amongst people who committed suicide. We can also see the distribution amongst the ages where people were diagnosed with depression.
All right, thanks, Jake, for that really interesting take on redesigning this genealogical tree. I know Lineage had its rectangular layout, and you chose the radial layout. One thing I was really curious about is the way that you chose to show those radial like bursts coming off the nodes. This is interesting for two reasons. Like in some of the phylogenetic data we talked about earlier, sometimes people use a pie chart. And then there was also the difference in Lineage where they kind of used it as a table. I really like the radial sunburst. I thought that was really neat. Could you tell me about the motivation behind that and why you made that particular design decision?
One thing that I wanted to do was make something that just jumped out to the user. So you can see that the radial trees, you can visualize the entire family structure at once. And then when using the different colors for the radial charts, you could see the clinical attributes for each individual. So you can get an overview of what the clinical attributes distribution was for each particular family.
Yeah, I really like that. Do you think that there might be some challenges as these trees start to get more dense? Have you thought about what you might do to help your approach scale? Because I totally agree with you. It really sort of sticks out what's happening.
Yeah, one thing that definitely needs to be integrated in this visualization is aggregation. So with use of the bigger families, the structure of the radial tree is set to a particular diameter. And so it gets really crowded with the bigger family. So aggregation to only show the important individuals would definitely be needed to be integrated.
Awesome. Thank you so much for that, Jake. That was really cool take on this.
Thank you.
So we are now going to actually switch gears from our data challenge to our redesign challenge. So Joy, this concludes the question and answer portion. And so the first talk that we are going to go to in our redesign challenge is an enhanced MA plot with R-Shiny to ease exploratory analysis of transcriptomic data. And I just wanted to give a brief context about our redesign challenge. It's an open challenge to the community, and it just says, pick a visualization that you think might need a different think through and tell us why that is and how you would redesign it. So if we could go ahead now and start with Michel Aupetit's talk on his redesign challenge.
Hello, I'm Michael Aupetit, Senior Scientist at Kata Computing Research Institute. I will present you an enhanced MA plot with R-Shiny to ease exploratory analysis of transcriptomic data. This is a joint work with Ali Sheharyar from Texas A&M University at Qatar, Talar Yacoubian, Dina Al-Jogol, Borbala Mifsud, Dina Al-Thani from Hamad bin Khalifa University. So we interviewed 3 expert users about tools they use for analysis. A PhD student in genomics and precision medicine, an assistant professor in health and life science, and a PhD in genetics. And from this discussion, we decided to focus on the MA plot. So an MA plot is a static scatter plot, typically used in RNA sequencing data analysis. A differential gene expression read count data are displayed between 2 conditions, C and T, for instance, control and treated samples. Each point in the plot is a gene. The x-axis shows the average expression level of the gene between C and T, and the y-axis shows the fold change between C and T. The color codes for the significance and the sign of the difference based on the p-value. So the Express needs to ease analytic tasks based on this plot. So remind this plot is not interactive, it's static for now. So for instance, they want to know what genes correspond to some specific points in the plot. They want to know where specific genes are in the plot. They want to detect genes which have specific parameter values. And they would like also to easily import and export genes from or to other tools like HiGlass or ShinyGo to get geo-annotation or study these genes in IC in tools exploring IC data. So we designed this dashboard with R-Shiny. So R, the most used language in bioinformatics, and Shiny is a toolbox in R package which allows to develop interactive visualization that can be opened in the browser. So in this dashboard, the main central part, of course, is the MA plot we use frequently, which allows for standard pan, zoom, and lasso interactions. And the rest of the dashboard is organized this way. So we load the data on the top left. The data are displayed. Then we can select some data using the lasso tool. These selected genes appear in this box. We can also import or copy-paste other genes in this box, and we can combine the two sets of genes with these buttons. We can filter based on different parameters, and we get immediately the result in this box. Here, either we are happy with the result and we can export these genes or copy-pasting these genes into other tools, or we can track them by pushing that button. Tracking means they appear, they are highlighted in this plot to allow for refinement, so we can either select some of them which has been highlighted, or we can look for genes around these specific genes, and so on and so forth. For more rational, so we use the diverging color scale for encoding the p-value, because we want to encode the significance and the sign of the difference to information. For the filters, we use very standard sliders and checkboxes. And for text area, we use text area to import, search, and track genes. We also organize the dashboard to facilitate this cyclic interaction, where we first import data on the top left. We then display this data. We select and filter at the bottom left, and we export at the bottom right. So if we are not happy with it before exporting, we can track and refine the search in this cycling pattern, and then export only at the end. We perform a summative evaluation with user A and B. C was not available. So we run a system usability scale, and we get 95% score, which is excellent for user A, and 65% of usability score, which is fair for user B. The main point was the tool is easy to learn and use. The negative point was missing information of required data format and pre-processing at the time of the study. But since that time, we updated the version, so now we display tooltips and give information about this data format to the users. For usefulness, A and B confirm the usefulness for exploratory analysis. They are willing to use this tool for their own projects, and they recommend it to other users. Thank you for your attention.
All right, thanks for this interesting take on the redesign of some RNA data. The thing that really struck me was the way that you surfaced that process, that repetitive process, and integrated into your dashboard. Could you tell us a bit more about that and how you linked it specifically to these different zones when you were developing it?
So you mean the full dashboard, the architecture of the dashboard itself, right?
Yeah, it was a really interesting architecture.
In the sense that we remarked that the user wanted to loop, in fact, to refine the search.
That's right.
It was not enough just to select genes. It was important to be able to re-inject the selected genes into the process to refine the search, to expand or to select, to refine the selection. So that's why we use this, and we were looking for how to design this loop in this dashboard, which is kind of rectangular, and we follow this Z pattern where we have the input on the top left and output on the bottom right usually.
Nice.
And we just have this standard Z pattern, but the filter happens on the bottom. And when we want to track, either I mean, the result appear here, so I can just show you here. The result appear here, and we can either save this result some files, or we can just select these specific genes to track them, so they will appear in green, and do other selection on top of that. So we can do refinement and search around the specific genes that we track, copy paste these genes. So either from the box here, we can copy paste them in some other tools, or we can just focus on them and copy from other tools to that specific input. So that's just a tool that facilitates this, basically. So it's a tool to ease the interactions.
That's fantastic. And was the Z pattern something that you'd seen before in other places and chose to emulate, or was it something that you felt made the most sense for this iterative search that you're supporting here?
I mean, it's a well-known pattern in this, when you design dashboards in interface or when you read text and so on. But I did not specifically see it in other visualization, but I thought here it was natural to use it. So, yes.
I think so as well. All right, thank you very much, Michelle.
Thank you very much.
We will now move on to our last challenge submission. This is from Romain Villemont, and he is going to be talking about the sawtooth chart for the compact cumulative data visualization.
Hi everybody, my name is Romain Villemont, and I'm going to introduce you to the sawtooth chart, a redesign of the cumulative line chart. It all started with this now infamous cumulative line chart of COVID-19 deaths. It basically represents the total number of deaths by country over time. I addressed 3 shortcomings of this chart. The first one is related to the visual clutter in this area where countries have reached the same total number of deaths. The second challenge I address is the growth rate comparison. Here it's difficult to compare the US and Brazil. And finally, I address the magnitude issue. This chart tends to be very abstract, and people tend to forget the total number of people that have been impacted. How did I address those 3 challenges? Regarding the visual clutter, I used the same design as horizon graph. That means I split the space into rows, and I put the countries in each row and make the country fit in each row. I use the slope charts by comparing time intervals instead of every single time point, and the trend will be visualized as the slope of the segment that connects those 2 time intervals. And finally, I used unitization techniques, basically by dividing large value into smaller ones. How did I apply those techniques to redesign the cumulative line chart? So first, I defined a unit, here 500 deaths. Then, every time this unit has been reached, I recorded the time interval. And for each time interval, I simplified it using slope graph. I then align the slope graph, and then you get the sawtooth chart. How do you read this chart? Well, you read each segment, and it tells you, based on how flat or steep it is, how much time it took to reach 500 deaths. Here it took a couple of weeks. And unfortunately, the segment slope got steeper, and that sometimes only took a couple of days or even less than a day to reach 500 deaths. I'm going to briefly talk about a difficult data processing challenge we had to address. Basically, it's related to the fact that daily reports didn't match our unit system. Like here, the report for this day was 700 deaths, so we had a first segment and another one, but how do you define when during that day 500 deaths have been reached? So we made this strong assumption that deaths were actually uniformly distributed during the day, which is actually not true, but that helped us to divide a single day that had more than our unit into our unit system. If you want to learn more about this process, you can read this observable notebook by Philippe Rivière that details it. Here are some examples of application of our technique to many countries. First, we use the sawtooth chart to align the countries by the first 500 deaths. So here you get how many days it took to reach 500 deaths for each country. And we also use the sawtooth chart to identify irregularities in growth rates and very high increase in the growth rates like the US back in April. How did I design the technique? I use an open and transparent approach by regularly publishing as a Twitter thread all my design iterations, and that enabled me after 15 days to get to the final version. During that day, I got many feedback from the Twitter community, got some feature requests that actually were very helpful, and I implemented many of those. There was this interesting discussion around the name I should use, and I ended up using the sawtooth chart as a reference to the sawtooth signal in signal processing. And finally, of course, there was lots of criticisms, but most of them were related to the dataset and the countries I was picking for my visualization. All the code and data for this chart are available online. And that's it. Thank you for your attention.
Thanks for that really interesting talk, Romain. I really liked the sawtooth chart. I thought it communicated this really critical and important thing about the outbreak, COVID outbreak, which is it getting worse, and how much worse is it getting? I want you to talk a little bit more about some of the reception that you got on these sorts of visualizations. Did other people find it helpful? Did they find it alarmist? Was there something in particular about the design that they focused in on?
Yeah, thanks for the invitation. Well, I think most of the debate was really about the data. I think like there was so many discussion and about like the countries I picked and actually the metric I was using and the alignments should align by first depth, first 100 of depth. And but I think since then I haven't updated the chart because it's broken now. There's been too much increase, and I really need to I think relook at the design, especially the unitization. I used 500 depths at that time. I think that was the way to kind of neatly break down the cumulative line chart. But now I need really to revisit it and probably pick a different unit.
Well, that's a terrible thing to realize, that there are so many deaths that you need to redo your unit. I had one more question in mind for you. Okay, see the observable stuff. Okay, and as you are thinking about scaling it up, is there one particular direction that you think might be more fruitful? You know, you mentioned the time unit, but maybe even, you know, is there something else in the visual design that you might change, unfortunately, as the outbreak gets worse?
Yeah, that's a really good question, and I don't know. It's again, I think it's really about the type of data to use. I would probably, I don't know, pick a different data set, but I think really most of the discussion and the debates were around the data. Around the design, there was not that many discussions. I think nobody really mentioned the unit I've been using. Like 500 seems to be fine, but yeah.
Okay, yeah, the underlying data is a common theme that we've talked about today, whether it's combining it or picking the right metric for it, putting it all together, the data is the problem. All right, thank you, Romain. I really appreciate it. And so we're now going to go on to our final bit of BioVis, our capstone presentation. I am really delighted to welcome Niels. As I mentioned at the very beginning, Niels was one of the founders of BioVis, and he's going to tell us a little bit about how BioVis has changed over the years and what his perception of just being a part of this community has been like. Right, so With that, take it away, Niels. Go ahead, Niels.
Hey everyone, thank you again for the invitation to be here today to talk to you a little bit about the last 10 years of BioVis and possibly the next 10 years of BioVis. And what I want to share with you today, as Anna said, is sort of my personal view on BioVis. Given that I only have about 25 minutes to talk today, I didn't even attempt to try to squeeze everything BioVis is, which is a lot, into this presentation. So what I'll talk about is sort of my personal view, my personal experience in this community, with this conference, and with this field of biomedical and data visualization research. So I want to offer you to maybe for a moment reflect on why you're here today. There's a cell on the screen right now, and that cell represents life and biology and maybe disease, health, and similar related topics. And in my mind, those are the reasons why we care about visualization of biological data, which this meeting and this community is all about. Because we have an interest in understanding life and understanding how biology affects human health and disease. And as you will hopefully sort of see, as I try to sort of create an arc from this single cell all the way to the end of my presentation, when we look a little bit into the future, I want you to keep that in mind, like that the reason why we do this is because working on visualization of biological data can have a real impact on people's lives and their quality of life. And I think I don't need to remind anyone here that I am here in my living room and not in Salt Lake City, because we are currently sort of struggling with one major and maybe the biggest health crisis that our generations, the people who are here, have ever experienced. And hopefully we won't experience anything worse than what we're currently going through. Of course, COVID-19 is also, as we've just seen in Roman's presentation, providing us with more opportunities to apply the expertise and the knowledge of this community to make things better. And with that, I want to give you a quick overview of what I want to cover today. And maybe not surprisingly, it's pretty straightforward. I want to talk a little bit about the past of BioVis, the last 10 years, how this all started. I want to talk about the challenges sort of that we try to address over the past decade, and some of which, to be honest, we're still working on. We haven't solved all of them. Then I want to talk a little about the present and what we're doing at the moment, what is happening in this field of BioVis, and what is its impact. And finally, I want to talk about the future. So straightforward, and let's talk a little bit about the past. So for those of you who are at BioVis for the first time, it's been, as I just mentioned, been a decade since BioVis launched at the VIS conference. Back then it was InfoVis. We launched it, or it was, what was it called? It was VIS Week, I think, the first time we had it in Providence, Rhode Island. This was 2011. when the first BioVis meeting was held. And I remember it being in this enormous lecture hall, and the room was actually pretty full for most of the sessions. So this was very exciting. Of course, BioVis didn't just start overnight. So there was a whole year before that. And actually, it really started in Salt Lake City, where we would be today if we wouldn't be dealing with COVID. It was in Salt Lake City in the Grand America Hotel in a windowless, literally windowless, meeting room for a breakfast meeting where I was invited to join a couple other people from the community to talk about whether we could create a new workshop or meeting around this topic of biological data visualization. I also do want to point out there are too many people to thank and include in this presentation, so I will pretty much not mention anyone. I think all of you who've been involved in BioVis for the past decade know what you've done and how much time you've invested in it and how much of your energy went into this meeting. I appreciate all of you, and without you, it would not have been possible. But before I start listing names after names after names and then risking of missing half of the people who've contributed, I decided I will leave the names out. So, but we started this in 2010, talked about, what could a meeting like this look like? And also how could this meeting, really be distinct from other meetings that had happened in the same space. There were some biomedical and really more medically oriented visualization workshops and meetings. There was, of course, a VisFCAC meeting, which many of you will be familiar with, which was sort of more driven out of the biological or computational biology community with a different intent. VisFCAC was always meant to inform more about what is the state of the art. As you know, it's not a meeting where you can submit papers usually. It's a meeting that inspires people to think more about visualization, really shows sort of the best work in that field. While BioVis was designed to sort of complement the VisFCAC meeting, be more focused on driving cutting edge research by inviting contributions of papers, full-length papers initially, and now we've moved a little bit away from that in order to really move the field forward. So, and then, going back to 2011, this is when it all started, and it's been going ever since. Initially, BioVis was at the VIS conferences, and then went to the ISMB conference, which is a conference hosted by the National Society for Computational Biology, came back to VIS, and ultimately they decided to move the main meeting permanently into the ISMB conference when that conference was reorganized and made room for meetings like BioVis. And BioVis has now become sort of a staple at that conference where it really draws very large crowds that attend the meeting every year. So after having talked a little bit about the birth of BioVis, I do want to spend a few moments reflecting on the first keynote at BioVis. And so that keynote was given by someone called Chen, who's a cancer researcher. And Linda Chen spoke about the Cancer Genome Atlas project. And the Cancer Genome Atlas, also known as TCGA, was for me one of the first large-scale team science projects that I was involved in. And it was also overall one of the first NIH-funded team science projects that was meant to bring together a very large community of cancer researchers, computational researchers, clinicians, and others who were tasked with essentially creating sort of a molecular map of cancer, many different cancer types across many different tissues. And the study in the end involved thousands of tumor samples, normal samples, in some cases metastatic tumors as well. And the number of assays that were applied to the materials collected to the specimens was actually fairly small. There were some sequencing-based assays, initially even some microarrays for gene expression data. There were a little bit of proteomics data that was generated with also array-based technologies back then, and whole exome and in some cases whole genome sequencing, and a little bit of clinical information. And that project had an enormous impact. The papers that were published directly out of TCGA and then follow-up projects really have driven cancer research into a completely new direction where these kind of large-scale collaborative efforts are very, very common. And that is something that I believe has really allowed the field, like these kind of large-scale team science projects really have allowed BioVis to blossom in certain areas. This is not all the areas where BioVis is blossoming, but this is an opportunity. These large-scale consortia or have provided opportunities for many people who work on visualization of biological data to get their tools in front of larger user audiences, which, as you know, for most visualization research, when it's in an application domain, is really, really critical that you get users for the tools that you create, for the techniques that you design. And so these consortia are extremely promising. And I want to come back to that in a little bit. But that keynote was certainly inspiring in that it presented the first BioVis crowd with lots of unsolved questions about extremely large and complex data sets. And well, you know, what did this really lead to was sort of that we started working on various challenges, and this field really had to address questions related to data. As I've heard, as I joined the session, you know, getting access to the data is actually really hard. Getting access to the infrastructure is one of the biggest challenges that you need to overcome, and this is a little bit of a theme in this presentation as well. Another issue related to data, of course, is the scale of data sets. They are definitely not small. In most cases, biological data sets these days contain gigabytes or terabytes of data, or if not hundreds of terabytes of data, and you're going into spaces where there's imaging data, often multiplexed high-resolution imaging data, or a large sequencing data sets. So these kind of challenges are challenges that we started working on in the past decade and started to think about how do we address this. And I will give you a couple of examples in the next couple of minutes of where I think we succeeded, and we'll point out many other problems where we haven't succeeded yet. The second point here is integration, and data integration is probably the #1 reason for why we use, or why we want to create visualization tools, because visualization of biological data is a very straightforward way of integrating data without spending too much time thinking about it. And that's also one of the reasons why it's so hard sometimes to create biological data visualizations, because in order to interpret biological data, most people want to bring in a lot of other information, other data that would help them understand the patterns that they're seeing in the data that they just generated. So the annotation of, say, genomic data sets or ontological information, if you're looking at, say, a transcriptomics data set, and you're trying to understand, like, what does it mean that the set of 25 genes is changing their expression levels or its expression levels. That's something that requires access to external data usually, and to bring that in is really critical. And finally, one issue that we started thinking about and still are thinking about very hard, user experience. And here I want to spend a few moments talking about user experience for tools for biological data visualization. So this roll duct tape is an excellent example in my mind to explain the state of the art of tooling in bioinformatics and computational biology. Most tools that are being used are just sort of patched together with a little bit of glue. Often this is sort of simple scripting languages or more advanced workflow languages that hold together different tools that need to be executed in order to get certain results. They also often sort of just are not hooked up together at all. So there's a lot of manual intervention where data is being moved from one tool to another. As long as the file formats align, you can do that. If they don't align, well, then there's another conversion step that's needed. And that is probably the bread and butter of many bioinformaticians out there who just need to worry about getting their data from one format into another in order to move it to the next tool that they want to use in their tool chain. And so that makes it, in my mind, really, really hard to improve the usability of visualization tools or any tools that use graphical user interfaces in biology, because there is so much patching happening. And another challenge that we're facing is that when you're building tools for exploration and hypothesis generation, they will make themselves obsolete essentially after some time. Like once we have enough understanding of the questions we're trying to answer with the data, we can hopefully learn enough from these visualization tools that we are able to create tools that allow us to automate the process. So moving from purely human interaction with the data, maybe enhanced with some algorithmic approaches, we ultimately, of course, want to move away from the visualization, and we want to move to automation and to create algorithms that can solve these problems for us. Otherwise, we're going to be stuck with a simple issue that will take a lot of human resources, but could be solved computationally. So that's an issue that we're facing, which is also an opportunity, because it means there's always going to be a new opportunity to create another visualization tool in the future. So science is also a bit challenging. So we're creating tools in many cases in BioVis to drive scientific progress. And scientific progress is not about creating hardened infrastructures that can be built on. A lot of science is as I say here, is trailblazing. It is moving quickly. And we try to come to some biological insight, to some understanding of like what is going on, and then we move to the next stage. And that makes it hard, again, to build tools because the infrastructure is often lacking. It's often very brittle infrastructure. And yeah, tool development often is challenging when you're trying to keep up with what sort of experimentalists are doing and how quickly the field is moving. So it's also hard to fund to build infrastructure. That's something that is well known for anyone who's tried to. Sustaining software development and tool building and tool maintenance is not straightforward. There are not a lot of programs that explicitly fund that because it's not considered science, it's engineering, and it's boring to the funders in many cases. And on the other hand, it's absolutely essential for biomedical research. And what that really leads us to, and understanding that has become obvious and clear over the last couple of years in BioVis, is we really need to think about modular approaches that we can reuse in different settings. That is true for the visualization components of these visualization tools, but it's also true for the data access components that we need, right? So we are dealing with many different databases, file formats, etc. Having modular components allow us to read data from different sources, and being able to put them together quickly with user interfaces is really, really critical and something we need to think about more, and we need to build more of. But again, this is infrastructure building, and again, harder to fund and to maintain than other things. Okay, here's another challenge that we've been facing during the early years of BioVis, which I think we're luckily now at a point of which we're realizing that has to change. And that is sort of the bulk nature of biological science and computational biology. So I'm showing you this letter here as an example, right? So think about these transactions that are happening in biological research, and the data processing is something like a letter, right? You write a letter, you send it to someone, it takes forever, it eventually comes back, they will read it. But what we're after and what visualization needs are much faster ways of interacting or moving information between different types of tools and between different approaches to analyzing data. So being able to go quickly between bulk process of data in a pipeline to an interactive visualization tool is something we really need to address. And that's that kind of nature of how a lot of biological research is being conducted is one of the reasons why it's hard to engineer good visualization tools, right? So batch processing and pipelines have been the focus of many of these tools, like in particular genomic data analysis is essentially all pipelines and batch processing. They are not designed for interactivity and user intervention. And I mean, we've said this, many people have said this in the last 10 years, but in visualization, interactive visualization and computational pipelines and computational approaches, they really have to go hand in hand, and they have to be designed together, because retrofitting these pipelines, again, it's just infrastructure work that someone has to do. And usually it's the visualization designer who has to do that, or the visualization developer, because, well, the pipeline's already there, it's generating results. And if you want to visualize it, well, then it's your problem to figure that out. And a lot of that has to do with file formats that were typically not designed for random access. or to contain, for example, aggregated data, which again is not needed if you're just doing batch processing. But if you're trying to interact with large-scale data sets, you need to have aggregated subsets of the data in order to visualize them at a reasonable speed. And that has started to change, luckily. And so now we're coming to the present. And this is something where I want to spend a few moments talking a little bit about how BioVis is going mainstream. I want to talk a little bit about opportunities and then the new exciting technologies that our colleagues in the experimental labs have come up with. And so BioVis is going mainstream. I want to keep this short, but it's really critical for those of you who are here who are maybe thinking about starting a career with a focus on biological data visualization. There are funding opportunities. I can only speak about NIH and the United States at the moment. I didn't do the research to look into how this looks in other countries at the moment. But in the United States, the National Institutes of Health have put out several funding opportunity announcements that explicitly ask for data visualization expertise as part of larger consortia, or even specifically for the visualization or development of visualization tools, for example, in cancer and cancer research. And that is new. Several funding agencies had visualization or biological data visualization as part of sort of their mission in their sort of roadmap documents, but there was very little funding. This has changed significantly over the last 5 years or so, where there's really an understanding on the side of the funders how important data visualization has become. And that is the first step, I think, in establishing a field like BioVis for the future. I'll come back to that in a moment. Now, opportunities. So as I mentioned earlier, these large consortia, in my opinion, and this is really just my opinion, if I talk to my colleagues and maybe people who are a bit more senior than me and who are worried about my career, tell me, oh, why are you being part of so many consortia? That's a lot of teleconferences. And yep, now life is a teleconference now, so I'm not too worried about that. But the opportunities that large consortia are providing in terms of interacting directly with the tool developers who are, or the developers of algorithms and computational tools, and those who are building the infrastructure to support these large-scale projects, is a really unique opportunity to get visualization built in from the start. And I will move on in a moment to show you an example of one such or two such consortia where we've been able to sort of get a foot in the door when these projects started. And I want to show you what kind of visualization tools we're able to build because of that. So the new technologies, we'll come back to that in a moment. But here is an example of a visualization tool that my lab has developed. It's called HiGlass. It's a framework for genomic data visualization, which can visualize genomic interaction data in these matrices that you're seeing here. It's interactive, of course. You can see it right here. And what we're seeing are interaction matrices for two different conditions on the left and right, and then top bottom is just overview in detail. No need to understand any specifics here, but what is really important to understand here is that this visualization software, HiGlass, is built directly into, among other places, the data portal for an NIH consortium called the 40-Nucleum Project. And because my team was involved in the design of the data formats that are being used to store large genomic interaction matrices, this allowed us to design them in a way that they're compatible with both computational pipelines as well as with visualization tools such as HiGlass. So that allowed us to really remove several barriers on the way from generating genomic data sets and visualizing them. So users can now look at a data set in the data portal, which I would show you if we had more time, but they can go there, they can look at the data that's available, they can see the data set provenance, they have all the information available, but they also have interactive visualization tools like this sort of multiple linked view setup available directly in that data portal. Now, this was 40-Nucleum, which essentially has genomic data. There are some imaging data, but that's not really playing a major role at the moment. Now, what has happened though is that there are many new consortia that are making use of technologies that were not around 10 years ago. Even 40-Nucleum is using technologies that only sort of became really available sort of from the early days of the BioVis conference onwards. But now what has changed is that we're now dealing with, rather than bulk data sets. So bulk means you take a clump of tissue and extract, for example, DNA out of it, and try to make sense out of all of these different cell types that are in this tissue piece together, which is not, I mean, this has led to a lot of new insights, but it's not really sufficient to fully understand biology at the single cell level. And that is where really the opportunities are abound for data visualization, is single cell data. And whoops, when I go and show you this little video here, if I hopefully play it. This is a visualization tool called VIS, also integrated in the data portal. In this case, it's the HubMap, the Human Biomolecular Atlas Program data portal, which is another consortium that we're part of, where we're dealing with both single cell and single cell information as well as spatial information. So we're not only looking at, say, gene expression levels across thousands of cells, we also in many cases have their spatial context. And we're not only dealing with sort of genomic assays, but we're dealing with imaging assays, we're dealing with transcriptomic assays, we're dealing with proteomic assays, metabolomic assays. And most of them are brand new, have only sort of been developed in the last 3 or 4 years. They are extremely unique in many ways. And this HubMap project is trying to put all of that together in order to build an atlas of human tissues, healthy human tissues at the single cell level. And what that means is we have to create visualization tools that can bring all of these different data modalities together in a single interface or single interfaces, because ultimately we want to navigate, say, from a transcriptomic data set to an imaging data set where we find the same cell types in their spatial context. We want to understand, for example, what is the composition of cell types in a certain tissue, and then look up how these cell types cluster together when we look into other tissues. And all of that makes it tremendously challenging to build visualization tools because, again, we're dealing with these different data types. And again, the importance here is to be involved early on when these consortia launch, and to be part of the people who build the infrastructure to make sure that visualization has its place. And what you unfortunately are hearing here is that we're doing an awful lot of engineering work. It's really a lot of software development. It's a lot of sort of very basic work in just getting tools to work. And I want to show you one example here. So this overview here, this figure, which I will not go into any detail in, there's a preprint or there's a website, viv.gelnborglab.org, is a visualization library for imaging data, for multiplexed imaging data. So we have high resolution microscopy images, for example, stored in OMI-TIFF files, 50,000 by 50,000 pixels. But then we have, for each pixel, we have a dozen to several dozen channels, each corresponding to a specific antibody that are contained in these data sets. And we want to visualize that on the web. Usually that means either you have to pre-generate some lower resolution images, pick 3 or 4 channels, create a JPEG file, serve that out in OpenSeaDragon. But that prevents you from actually exploring the data sets because you cannot pre-generate all possible combinations of channels. Alternatively, that's the middle row here, you could use a server such as the Omera server that would allow you to change channels on the fly, but then you need to maintain a server infrastructure. And so what we've done, we've developed this library that can render these kind of images, highly multiplexed, high resolution, high bit depth, OMI-TIFF files and ZAR files directly in the web browser. So you can check it out, it's fun, it's very interesting to look at these images. Very little data visualization research happening here, but it's an essential part for us to bring data visualization tools for imaging data that can be integrated with other data types into data portals like the HubMap data portal. As I just mentioned, ZAR is a new file format. It's coming out of the Python community. It's extremely useful to store so these multidimensional arrays can be used for data visualization. Okay, so the future. I want to keep it short because I think we're running out of time. I'm not exactly sure when I started and how much time I have left. But I want to talk a little bit about what's coming for BioVis. So let's have a quick look at that. My first point is that clinical and medical applications of sort of traditional BioVis data, meaning genomics data and other molecular assays, is becoming more commonplace. And there also has been a lot of progress in terms of making clinical data accessible to tool developers. For example, if you're thinking about electronic health record data, for those of you who have an iPhone who use the Apple Health app, Apple Health is using an interface called Smart on Fire, which uses standardized interfaces to electronic health record systems. And these electronic health record systems, of course, can contain an awful lot of different data, including genomic data. And with these kind of platforms, we're at least addressing this good old classic of how do I access data? And of course, if you think about it in the context of clinical data, there's not only how do I get to the data, but how do I actually do this in a secure fashion, right? You cannot just download a bunch of electronic health records and store them, on your web server and make them available there. This is certainly not going to fly. And so in that case, and this is also true, of course, for a lot of genomic data sets, even when you do some basic science, when they're human data sets, you need to protect them accordingly. And so you're dealing with an awful lot of authentication and authorization questions, as well as sort of data security that, again, will require you to work very closely with people who build the infrastructure in order to provide access to the data. So these clinical and medical applications are one of the new topics that I will be we will be talking about. And I think it's time for BioVis to re-engage in these conversations with the medical and clinical visualization community to make sure we're not losing track of each other's efforts. And the next issue that I briefly want to touch upon is the question of how do we sustain and grow this community? So education plays a huge role, of course, in a situation that this field is in now. There is funding, there's data, there's some people, but how do we get more people? So I'm running a research lab at a reasonably well-known institution, and believe it or not, it's really challenging to find postdoctoral fellows, PhD students who have the right skill set and the interest in dealing with data visualization. So what is the challenge that we're facing here? One is for those who are sort of coming out of traditional computational biology and bioinformatics programs or have that kind of training, often are lacking sort of this exposure to humans as part of the research. Right? They will be excellent engineers and excellent in algorithms, and they will know the data really well. But sort of this human-centered work that is central to BioVis is something that people from that community are often lacking and need to learn. On the other hand, for people who are coming more out of traditional, say, maybe more HCI related, or say someone with a pure visualization background out of a computer science program or even a computer science PhD, will have the right understanding of learning from humans, from users, integrating that into the research and design process, but often lacks enormously in terms of an understanding of the data that we're dealing with, which is really, really essential in terms of building BioVis tools. The data formats that are out there, the databases and resources that people often need to be familiar with in order to pull together sort of visualization tools that allow people to really interpret new and exciting data sets. The number of these external resources that need to be brought in is really daunting. And here is really, I think, an opportunity for this community to think about, what can we do to make sure that, going forward, there will be experts who know what BioVis is all about, who really have this understanding of computational biology, bioinformatics, data visualization, HCI, and are also, at least for the work that my group is doing, have to be really good engineers, because we need to build tools that fit into these ecosystems. I think I sound like a broken record when I repeat this, but it is really all about getting access to the data. And this put together really, in my mind, raises the question, how do we create these experts? Who will take that on to make sure that there is a growing community of BioVis researchers? And with that, I want to go back to this figure, and I know I have to wrap up. It's all about the human experts, the human as a user of these tools, and ultimately also the human as the target of this work. Right? We want to improve human health by developing tools that help us understand biology better. And then just a quick shout out here, or a quick note, I'm hiring some of these experts. If they're out there, if you're thinking about doing a postdoc starting now or in the next couple of months, let me know. We have several positions that are advertised. There are additional positions that are not advertised. Just reach out to me if you have the skills that I've talked about here in the last couple of minutes. And maybe you will be joining us in my lab to work on BioVis. And with that, I will wrap up. Thank you very much for your attention.
Thank you, Niels. So this brings us actually to the conclusion of BioVis, right by a hair, in fact. I really want to thank all of the people that presented today for joining us. And I hope that everyone will join us again next year for BioVis, hopefully in person. Thank you very much, everyone.
Digital humanities present great opportunities for testing new visualization approaches and evaluation techniques. However, and given the diffuse character and novelty of the field, it may also be intimidating for novel and senior researchers willing to get started in the discipline. In this paper, we propose a data-driven analysis of visualization for